Now, he's just based, Dean Dorsey's going to come on and talk, and then I will talk. Okay. Okay. Good. Great. Yeah. Okay. Good. We're set. Thank you. And he speaks first. Good.
Well, good morning, everyone, and welcome to this colloquium for the RU program at the University of Georgia. Though I know it's been a lot of hard work, I hope that your work over the summer has been both stimulating and fun. I'm a big fan of undergraduate research. My experience with undergraduate research goes way back when I was a student to the summer of 1977, when I was able to participate in a summer research program for high school students in the Washington, D.C. area. I had the opportunity to work at the National Bureau of Standards in Gaithersburg, Maryland, what is now called the National Institutes for Standards and Technology, on a project that involved precision polarimetry measurements of sucrose solutions. The details are a little fuzzy now, but I do recall enjoying working in a lab alongside PhD chemists and physicists. My initial participation turned into an ongoing summer job. I was fortunate to work at NBS during every summer as an undergraduate student, working on a variety of projects alongside scientists and engineers. These experiences influenced my decision on a course of study in college, and I eventually majored in physics. I went on to earn a PhD in physics and became a faculty member at the University of Virginia and then the University of Florida. While at UF, I decided it was time to pay back the benefits I earned from my undergraduate research experience, and in 1999, I co-founded a summer RU program in UF's physics department. We would bring 15 students to Gainesville every summer to conduct research in materials physics. I served as the research mentor for several of the students, and it's gratifying that some of them are now faculty members at colleges and universities around the country. I occasionally bump into them at professional meetings and conferences. Why is undergraduate research so important? It's the authentic experience of discovery, the pleasure of finding things out, to quote the irascible 20th century physicist and Nobel laureate Richard Feynman. It's about discovering or learning something that nobody else knew, or at least they didn't know it in the way that you know it. It's learning about new scientific techniques, working in teams, and alongside some of our talented UGA scientists. It's presenting the fruits of your work in accessible terms to your peers and to others. It's also about dealing with failure. Scientific research seldom proceeds in a linear fashion, and it's always fraught with pitfalls and failed experiments, dead ends, and misconceptions. Sometimes more things go wrong than right, and practicing scientists develop patience, persistence, and perseverance, which are important life skills that you can well apply outside your scientific pursuits. And speaking of which, I know that many of you will be thinking about graduate programs soon. And as good students, I'm sure we'll have many options from which to choose. I hope that you'll seriously consider UGA as one of your options. As I'm sure you've observed, we have outstanding faculty and great facilities at the University of Georgia. Please stay in touch with your research mentor as you consider your options. If offered a spot in one of our graduate programs, I hope that you'll commit to the G. I hope your summer was productive and stimulating. I expect you'll have a better idea about the hard work involved in scientific research, and hopefully you've experienced the joy of discovery. I wish you all the best in your future academic endeavors. Thank you, and have a great symposium. And I'm going to turn this back over to Dr. Arnold.
Thank you, Dr. Dorsey. I really appreciate your support over the years, and it's really part of the success of our program and builds on your own experience. This program is also sponsored, as yours was, by the National Science Foundation, and we're very grateful for the 20 years plus of support from Sally O'Connor, who has been remarkably our program director now for the whole length of the program. I don't think that happens very often with an NSF program officer, but I am deeply appreciative, and I've seen all the new features she's allowed to build on the programs we have. And one of those features I'll talk about a little today. So this program started in 2000 under the sponsorship of Provost Karen Holbrook and Dr. Harris at Clark Atlanta University. Continues right down to this day to be a partnership with Clark Atlanta and the Atlanta University Center. And that's how the program began, and they funded us for a couple of years, and then NSF decided we were serious, and they continued supporting us down to the present. This program has been in existence long enough that some of the early participants back in, say, 2003 or 4 have now moved on to faculty positions or leadership positions in national labs. And so this also gives a unique opportunity for the current participants to reach out and see what kind of experience lies ahead in their future. I want to again emphasize that the key support for this program began with the university, has been sustained by the National Science Foundation, but has been strongly supported by Franklin College through much of its history and by the Institute of Bioinformatics. And I'm really grateful for all of their support over the years. What I would like to do now is introduce our first speaker, and she is an example of one of these new initiatives at the National Science Foundation. She is actually the first, Mariah Salceda is the first post-baccalaureate NSF REU participant. This is a new program that Sally started, and Mariah was the first one nationally. She's obtained her undergraduate degree from University of Maryland, Baltimore County. She graduated from there and then moved to San Antonio, Texas. She was encouraged to go on and research, and fortunately she took Dean Dorsey's advice and applied to the University of Georgia. And she was actually admitted, and she's now in the integrative life sciences program where she can choose from a number of different programs. But I think she's pretty set on bioinformatics. But she has the opportunity to choose a variety of paths. She is going to talk about the collective behavior of oscillators in a noisy environment, and it's my pleasure to turn it over to Mariah and hear about her summer research. Thank you, Mariah.
Thank you, Dr. Arnold. So good morning, and I hope everyone's doing all right. As Dr. Arnold said, my name is Mariah Salcedo, and I did my summer research with the Arnold Lab. And I will be talking about collective behavior of single-cell oscillators in a noisy environment. So the first thing I want to do is kind of talk a little bit about what collective behavior is. So it's a term that some of you may or may not be familiar with. What we're talking about is a form of social behavior that is defined by coordination of large groups, as well as the emergent properties of these groups. So there are many striking visual examples in nature, such as the flocking of birds, the marching of locusts, schooling of fish, nesting of ants, and the clock. And that's a concept that I'll be expanding upon a little bit later. So people can also engage in collective behavior, from forming riots to fashion trends. So interestingly enough, we have found that the cells in our body also engage in collective behavior, from neurology to immunological responses to cell migration, which can be important for things like cancer research. And there's a little figure talking about epithelial collective migration. So keeping that in mind, that our cells engage in collective behavior, I want to talk a little bit more about the clock. So what is the clock? They are single-cell oscillators that respond to light and temperature changes, acting in a collective manner. So they are controlled by a feedback loop of several key genes that respond to light and temperature changes, and genes that kind of kick the whole oscillation off. It's important to keep in mind that every single-cell oscillator marches to the beat of its own drum, so to speak. But when you get enough of them together, they are able to sync up. Even when you have two groups of cells that are not in sync, through collective behavior, bringing them together, they can sync up. So an important question is how this happens. So what we found is this behavior is potentially regulated by stochastic resonance and quorum sensing. For stochastic resonance, you can think of a radio static. There's a certain frequency with which that static allows you to hear the song more clearly. With that, we have the definition that stochastic resonance is the intrinsic noise in a system that helps with focusing a signal of interest. For quorum sensing, it's important that you understand that cell density can change the behavior of a biological system. It could be something like the response to less resources or anything like that. But regardless, there is a change in gene expression. And with that, we have our definition, regulation of gene expression in response to fluctuations in a cell population density. So the Arnold Lab has created a mathematical model that shows that stochastic resonance is needed for the oscillations to sync up. And this resonance occurs at a certain cell density. This concept that marries stochastic resonance and quorum sensing is called stochastic coherence. And with that, I have my research question. How do we experimentally test stochastic coherence? So that's a lot of what I worked on this summer, is trying to figure that out. And with that, our mathematical model of stochastic coherence focused on transcriptional bursting to understand the oscillations occurring. The process for experimentally testing this mathematical model will involve a microfluidic device, a lot more math, and a way to see live mRNA transcriptional bursting. So this summer, I worked on the imaging system that will allow us to do just that. So the way this imaging system works is you have these loops, the MS2 and PP7 loops with their respective coat binding proteins. If you add the loops to your gene of interest and the fluorescent protein to the coat binding protein, when transcription occurs and these two factors bind, you get a fluorescent signal. And that's kind of what you're seeing at the bottom of this image here. So this will actually allow us to look at live mRNA imaging. So with that, I have my research plan. So to introduce these loops, coat proteins, and fluorescent proteins, we will need to introduce plasmids into our biological system, Neurospora, using the CRISPR-Cas9 system. Our first set of plasmids needs a promoter, the stem loop, and the gene of interest, and a location to be inserted. So we chose CCG2 as our gene of interest because it is part of that previously mentioned feedback loop and lets us know that our oscillators are in action. So you can kind of see a little outline of what I would need there. Our second set of plasmids will contain a promoter, a nuclear localization signal, a fluorescent protein, and the corresponding coat protein, and a location to be inserted. So as you can see, we actually decided to keep both set of loops for this experiment with different fluorescent colors to kind of have an orthogonal approach and confirm our system is working. And this will actually additionally potentially help us cut down background noise. And if I have enough time at the end of this, I can additionally speak about other orthogonal methods that we're planning to use to confirm that our results are biologically sound. But I'll kind of just move forward for now. So with that in mind, our lab already has some materials from previous experiments that we were doing, and that would be the guide RNA for CSR1 and Cas9. So very important for using CRISPR. So this means that the materials that I need would be the guide RNA for CCG2, the guide RNA for His3, and I would need to create our donor plasmids. So our donor plasmids will be created using a technique called Gibson assembly. What this will allow me to do is if I have amplified and then isolated all the different key pieces of our plasmid, then I can just throw them into this master mix and they will ligate into a new plasmid. Since I have to isolate each of these pieces either way, I decided to try them in different combinations in case one works better than another due to something like size or any other factor. So our lab uses Neurospora. It's a beautiful fungus and it takes some time to grow and cross. And more than likely, we will need to make several iterations of our plasmids to be sure our results are biologically sound. And due to this, it makes sense to pilot our study in a much faster growing organism. We decided to pilot our study in E. coli to be sure that the stem loops don't accidentally block translation, essentially knocking down genes, and that the stem loops don't result in mRNA accumulation. Additionally, we want to be sure that we have a sufficient number of loops so that the fluorescence can be picked up in a microfluidic environment. In addition to all of those things, all these experimental techniques are very new to me. So I'd also like confirmation that there wasn't simple mistakes made along the way and we can make corrections in a matter of days or weeks rather than months. So using super competent cells and therefore bypassing our need for the CRISPR-Cas9 system, we created a more bare bones version of our experiment to mostly test the number of stem loops needed for that microfluidic environment and to be sure translation is actually occurring at this stage and I didn't accidentally knock down the gene. So this is kind of the more bare bones system. Again, this will be done using Gibson assembly, and I will be using different combinations of fluorescence and coat proteins. So due to delays with ordering my plasmids, they actually just came in this week. So unfortunately, I don't have any confirmation of current loop number that would work for us. I was able to use the time to work on the NSF GRP grant and make a website for collective behavior, but now that my starting plasmids are here that will give me the portions that I need to amplify. I'll be working a little longer in the Arnold lab before the semester starts, just to see how much further we can push this project along. And I have the plasmid maps, and I think I have enough time to go ahead and explain a little bit about how plasmids work. So with this, every plasmid usually contains antibiotic resistance so that you can grow them and selectively choose for them. This is tandem dimer coat proteins because we read that that would work a little bit better. So that's how we designed ours to cut down background noise. And this is with the fluorescence M cherry. Every plasmid also needs an origin. And what these little ticks are that have all these strange abbreviations, these are restriction enzyme sites. So if I wasn't using Gibson assembly, that would be something that I would use to ligate and make new plasmids from. So I have these guys, and then I have something incredibly similar with the MS2 loops. Again, these are our tandem dimer. And then lastly, I have our CCG2 and the respective stem loops. So that's about all that I have to show, and I can answer any additional questions.
Thank you. That was wonderful. You were busy setting up a complicated series of experiments this summer.
Yes.
And so the first thing I'll just ask while we're waiting for questions to come in, you can put questions in the chat down below, and I will read them and we'll present them on the screen so that Mariah can answer them. Well, the first question will come from the moderator. So what did you need in this project in terms of people with different backgrounds to do the study of collective behavior of single oscillators?
So that's a really good question. There's a little bit of feedback, so sorry if I'm a little awkward. I'm going to take my headphones off really quickly.
Yes, that's good.
So in order to get this project accomplished, we kind of need a team, and it's going to be a very big interdisciplinary team. So there is the portion that is actually creating this system. And we actually reached out and found a collaborator in Portugal where all they do is work with imaging systems like this, because it is something that is pretty cutting edge and very, very new. And it's never been done in neurospora. So looking at transcriptional bursting in live mRNA is a task. And so we reached out for collaboration with that. In addition to that, the mathematical modeling that we'll do later will take some concepts from physics. It's the ensemble model. So we have someone on the team, and Dr. Arnold himself also does a lot of this. So later on, when we actually get our data, it'll include that. The microfluidic device itself is an engineering problem. So we have faculty. He's a doctor of engineering, and he's in that department, Dr. Mao. So him and his team are great, and they've helped us out in the past with microfluidic experiments. So it takes several disciplines to look at a question like this. And even though it might seem kind of simple at the start, where it's just like, oh, we're just gonna, make these things touch and glow, there's a lot that can potentially go wrong, and a lot of people that are great for being resources and asking questions too. So
yeah,
physics, engineering, biology, math.
So how did you get interested in the math side of the project? Where does that come from?
I've always been really, really interested in thinking about math. it kind of started from a really, really young age where I've always just been interested in reading nonfiction. And I found a really interesting subset of nonfiction that talked about physics. So I started there and going to used bookstores, finding things, books on things like chaos theory and such, kind of made me want to learn a little bit more just about the math side of things. So I'm really, really excited about the mathematical modeling portion of this project. That's probably the part that gets me the most excited, honestly. So,
So we have a question from the audience. Can you briefly describe the different components of the model you're thinking about?
Sure. So when we're looking at stochastic processes in general, when we mathematically model those, you're kind of almost putting things into a box and you're looking at probability in a way. I think we have another presenter, Tony Zhang, that's going to be talking a little bit more about this later on in the symposium, and how you do a thing called random walks with a lot of models for anything that deals with stochastic processes. So I'll probably leave a lot of that for him to explain because it takes a little while. But when you think about stochastic processes, you can think of the most simple one being just like a coin flip, where if you think of each coin being flipped, it's ones and zeros. And you can tell me that there's a 50-50 chance because that's our probability, but you can't tell me exactly where those ones and zeros lie. So we're going to be using a lot of probability. When they made this previous mathematical model, they did something kind of similar to those ones and zeros, where they looked at transcriptional bursting as being on and off. And so we're going to kind of take that concept, and that's why we're really focused on that transcriptional bursting, because that mathematical model really focused on those like ones and zeros, and that helps us with setting up a framework that we can actually look at experimentally. So I hope that was enough detail without taking too long.
So we have one other question, Jim, from Tanya Rosario. How are you using the microfluidics? What's the design of the system to answer questions about oscillation?
Okie dokie. So microfluidics are something that we've used in the past in this lab. It's very cool because what we can actually do is isolate at the single cell level while being able to capture these very detailed images. So that's mostly why we're using the microfluidic device, because we can control the environment, we can control temperature, we can control light, and that's essentially our noise in the system. And that's something that in further experiments we'll actually be playing with those values to kind of see how our oscillators react. And an important thing with stochastic coherence is it's very dependent on cell density. So we can very easily control cell density in that microfluidic environment because it's single cell, so we can kind of just add what we need, and that helps us a lot with our ability to control what's going on. So
Dr. Kai-Zhang Yi has a question. He's in the genetics department. He said, I may have missed this, but how many genes are you tracking in your experiment to test their collective transcriptional burst?
That's a really good question. And currently we're looking at CCG2, and we're going to kind of pilot everything with that to make sure that we kind of understand how the imaging system works. And then we're also going to be looking into the freak gene. Both of those are big indicators that the oscillations are occurring and acting. So tracking those kind of lets us, I guess, almost start at the beginning of those oscillations and really see when they're kicking off. There are other genes involved, as it's like a feedback loop. So there's other genes that are more temperature dependent and more light dependent. So I guess in the future, as we're seeing our experiment, that might be something that we decide to look more into. But for now, with our preliminary information in the mathematical model that we have, CCG2 and Freak should be good. And looking at both of them is kind of almost having a bit of an orthogonal method where if we see one and we see the other one doing really similar things, then we know that what we're looking at is biologically meaningful. So that's mostly why we're focusing on those genes. But maybe when we start playing with temperature and light, we might decide that it would be interesting to look at the genes that are more specific for that too.
Well, thank you very, very much for a very interesting and wonderful talk. And we're very lucky to have you on board working on this interesting project with so many other faculty.
Yes,
thank
you,
much,
Dr.
Maria.
Arnold, and thank you, everyone. I appreciate having the time to speak.
So our next speaker is Skylar Gay. We also very fortunate to have her working with us this summer. She's from Virginia Western Community College. She's a rising junior, and she is going to be talking to us about the pandemic, and hopefully you'll find this of general interest. I think it's probably of interest to all of us. And so it's my pleasure to introduce Skylar, and please go ahead.
Well, good morning, everyone. It's great to be here with all of you today. And thank you, Dr. Arnold, for that introduction. So hello, as Dr. Arnold said, I'm Skylar Gay. I've had the pleasure of working under Dr. Jonathan Arnold and Michael Scarrow for the duration of this summer. Today, I would like to talk about the research that I've completed in the University of Georgia REU. So, without further ado, here is SARS-CoV-2 gene flow mapping, a cladistic study of gene flow among whole genome sequences of SARS-CoV-2. Since December 2019, there have been nearly 200 million confirmed cases of SARS-CoV-2 globally. Of these cases, we have whole genome sequencing data for about 5,000 of the earliest cases of SARS-CoV-2. Among these samples, we have observed genetic mutations that have been leveraged to investigate the prevalence of gene flow within the SARS-CoV-2 infections. Mapping viral gene flow will allow the science community to better understand the transmission of SARS-CoV-2 pandemic, and this could possibly help prepare us for future outbreaks and pandemics of this sort. A data set of whole genome sequencing data for 5,209 of the earliest cases of SARS-CoV-2 was arranged into 9 clades to quantify gene flow by identifying samples as being at least 80% from one locality or having at least 80% bootstrap support. The central points of each of these clades and the geographical outliers from each clades were established. The MAPS package was used to display a global Mercator projection to demonstrate geographical locations of gene flow. And the Geosphere package was used to implement spherical trigonometry on the Mercator projection to draw arcing connections from the clade centers to the outliers that were previously determined. The number of cases in each outlier location was illustrated by color variation in the shades of the arcing connections that were plotted. This is a Mercator projection that demonstrates the gene flow of SARS-CoV-2 pandemic. Major cities are plotted on this map, ranging from New York City, USA to Moscow, Russia. The large dots on this map represent the center of the clades, and then the small black dots on this map represent the outliers that were determined. Each of these arcs represents an inferred gene flow event from the clade to the outlier. The number of cases in each outlier location was illustrated by, of course, the color variation in shades, and you can get a little more information on that by checking out the legend in the bottom left-hand corner of the figure. There are 5 clades in Europe, 3 in North America, and 1 in Asia. As you can see, and I'm going to take a second and enlarge this so we can get a better view of Europe here. There are a total of 5 clades, and there's 2 overlapping in the United Kingdom, so it's a little more difficult to see in this point. But look at the density of connections emanating from this point. from each of the European clades to another European clade, or simply to the outliers of the surrounding locations. That really does show just how many connections and the gene flow events were coming from Europe. This is a bar chart demonstrating the number of connections from the clade centers to the outliers that were identified in each geographically oriented clade. Now, keep in mind when looking at this, each of these clades was identified as being 80% from one locality or having at least 80% bootstrap support. This chart shows the number of connections that were drawn from the European clades far outnumber that of Asia and North America. While the clades centered in Wuhan, China, the top clade you can see on that bar graph, it had 9 connections, which is the most out of any of the clades, like total. And then the total connections from the European clades was 31. And you can see, again, that really does outnumber that you would see from Asia and North America, but we can talk a little bit more about why that occurred later. I wanted to take a second to look at the clade 7 phylogenetic tree. Now, clade 7 is centered in Wuhan, China. This tree displays the lineage of the SARS-CoV-2 pandemic from Wuhan, China, ending in a Canadian sample on this tree. The phylogenetic tree, of course, begins in Wuhan, China, the origin of the SARS-CoV-2 pandemic. And the branches reveal samples all the way from an American cruise all the way to Canada. The samples in this tree come from the Americas, Europe, and Asia. These outliers were shown on the previously shown figure of the Mercator projection. So we took a look at the phylogenetic tree for the origin of the pandemic. Let's take a look at this phylogenetic tree. This is clade 9, the United Kingdom. And of course, it displays the lineage of SARS-CoV-2 pandemic from the UK. So this tree, you can see it shows branches to reveal samples from Belgium to England. The majority of these samples in this tree are European, but I want to note the African sample, the DRC, Democratic Republic of the Congo, and one sample in New York, USA, and one Australian sample. These outliers, again, were displayed on the Mercator projection previously shown. So let's take a look at the results from these projections. The percent of gene flow shown using the connection map is about 1%. The European clades had a total of 31 connections, the Asian clades a total of 9 connections, and the North American clades had a total of 15 connections. The majority of the European connections emanated from clades centered in or including Belgium, Germany, and the UK, with 31 connections from these 5 clades. I want to really highlight the fact that these were all kind of located in Belgium, Germany, and the UK. So the high numbers of gene flow from Europe are intriguing, considering the origin of the pandemic, Wuhan, China, which is in Asia. If Wuhan, China is the origin of the SARS-CoV-2 pandemic, why are most of the connections emanating from Europe? Well, to best answer this question, I have assembled this flowchart of SARS-CoV-2 transmission using the data that I talked about previously. So the sample's location can be determined by the legend in the top right-hand corner, and that identifies the continent of the location that is demonstrated in the legend. In the symbols. Each of the symbols were organized into rows, and each of the rows are organized chronologically with the dates that you can see on the right-hand side of the screen. After the data was arranged into clades by identifying samples as being at least 80% from one locality or having 80% bootstrap support, samples were organized by date. So I took all of that data, which ended up being about 700 points, and organized that all by date. So we can see the connections that would be drawn from the clade centers to each of those outliers. Samples that were previously identified as outliers were closely examined to determine a point of origin, which was connected to an earlier location on this flowchart. The high number of connections in or including Belgium, Germany, and the UK could stem from a delay in precautionary measures, including rules about limited social gatherings and social distancing, or limited personal protective equipment, such as gloves, face shields, and masks. This number of connections from Europe could also be attributed to the lack of information known about the SARS-CoV-2 virus and maybe improper communication about the possible risks of this pandemic. And that could, again, stem from a lack of information known or, bad communication because of the so much information that was unknown about this virus at the time. I would like to take a moment to thank Dr. Jonathan Arnold, Dr. Liang Liu, and Michael Scarrow. Of course, Dr. Arnold and Dr. Liu were the overseers of this project. And my mentor, Michael Scarrow, thank you so much for your continued support throughout this project and throughout this REU. And even, throughout my career as a scientist, I really appreciate that. And a huge thank you to the NSF grant that made this research possible.
Thank you so much.
If we have any questions at this point, I would love to answer them. And if I'm unable to answer them this time, definitely, I would love to hear from you. And please reach out so I can continue to answer your questions and talk about some further research.
I do. You have a question already.
Okay.
So, and the first comment is, great job. Have you considered how sampling and sequencing capability factor into which locations are outliers? So that's an interesting.
Yeah, absolutely. Thank you for that. That's definitely a great question. When we were looking at this, we did see that, I'll take, for example, Africa. There was two outlier samples from Africa, the DRC and Senegal. Mm-hmm. Now, is this, again, kind of answering your question, is this because of a lack of sequencing centers or places that had the technology to really sequence this data? Or is this because there were only those, cases? And that's highly unlikely. I think most of the lack of outliers or information that we have could stem from a lack of the sequencing capability in the areas that were displayed on the map. So definitely great question.
Mm-hmm. You have another one from Tanya Rosario. Did your data set not capture the outbreak in Italy that happened early in the pandemic, or was there limited gene flow from that country, possibly because of protective measures, such as closing everything down?
Absolutely. Could we take a second? I would like to bring up the connection map again, just so we could kind of talk about that. Right. Okay, so Tanya, that's an excellent question. Unfortunately, if there was not data provided for that, and I remember that there was limited samples from Italy, and that could have been that was not included in any of the clades that we found, or simply there isn't enough sequencing data to really make a strong judgment call about that. But again, as you can see, there are so many connections emanating from Europe, and I think that is just, a huge number of samples at that time. So possibly there wasn't enough sequencing capabilities at that time to capture the data from Italy.
Right. That's very good. Yeah, so what was the relative number of cases that were coming, say, from Asia to the west coast of the US versus the number that came from Europe to the east coast of or parts of the United States?
Yeah, absolutely. So we found that there was about 6 connections coming from Wuhan, China, to the US. Now, again, that's just because that clade is centered in Wuhan, but kind of the Asian clades, there was only about 6 connections in total coming to the US. But there was, over that, I think about 9 connections from Europe to the US in total. So that again shows just how much, or just how many gene flow events occurred from Europe to, the United States.
So what are your plans for the future now that you're about to graduate from Virginia Western Community College? What are you considering in the near future?
Yeah, absolutely. So as far as research, I am definitely considering, I'm in contact to get further data for this particular map. I do want to continue this in that flowchart that I demonstrated. I do plan to continue to find resources to expand that. As far as my education-wise, I'll finish out at Virginia Western this spring, hoping to transfer to a four-year school. Of course, I'm in Virginia, so somewhere in Virginia, possibly. I have not considered that, or I don't know, maybe Georgia. But yeah, I'm definitely looking to continue to finish out my bachelor's program and continue to a PhD program.
Yeah, Dean Dorsey will be happy for you to consider Georgia. So thank you very much. Thank you for the very interesting presentation. And you've been a wonderful part of this project. I'm very
Yes.
grateful and privileged to have worked with you.
Well, thank you so much. I appreciate this opportunity.
So I was just going to tell people a little bit about how we try to strengthen the connections of the budding researchers like Skylar. This week we had a former REU participant, Nikita Womack, who went through the program in 2003, but she is now a member of the Uniformed Officer Corps that serves the Public Health Service. And she has had one of the challenging responsibilities of looking out at the pandemic in California. So she has the opportunity to talk with Nikita and can probably find out some additional information that will help her on this project. So thank you again. And I will now introduce our third speaker before we have a break. And this is Ayana Holland. She worked with this summer Dr. Kai-Zhang Yi, who is in the audience. She comes to us originally through Clark Atlanta University, that partnership there, but she's currently enrolled in Trinity, Washington, which is right in Washington, I guess right in Washington, D.C. And she is going to, in fact, also continue this theme of looking at the bioinformatic analysis of RNA sequencing data that also Skylar was involved in another project. So I'm so glad we're going to hear about this, and I'm going to turn it over to Ayana, and I'm so glad you're here.
Thank you. All right, so hi everyone, my name is Ayana Holland, and my project is bioinformatics analysis of RNA sequencing data to identify differentially expressed genes. The background of RNA sequencing is a technology that can determine the nucleotide sequence of the RNA molecules and to quantify the expression level of each RNA molecule. Comparing RNA sequence from different samples will provide insights into genes, biological processes responding to the specific conditions. RNA sequence can also produce a huge amount of data that requires bioinformatics analysis. RNA has many different types, including mRNA, tRNA, and rRNA. What is my project? During the summer, my research project is to find the genes that have different expression levels as a result of the two different genetic backgrounds from microglia in mice. Researchers can learn from the RNA sequence about how genes are transcribed into RNA. When performing RNA sequence, genes have RNA sequence in the two different biological samples. We can identify genes that have different expression levels between the two. And I have multiple methods of this project. I was given microglia of two different mouse strains, wild type versus A5 knockout. We have 4 biological samples for each strain, and we have RNA sequence data from our collaborator, and we have performed bioinformatic analysis to identify genes which are active and specialized in a specific strain. So this graph that you guys see in the right-hand corner is a wide range of R packages were developed for this purpose, such as EdgeR, Glemma, and Lemma. After bioinformatics with various packages in RStudio, we will identify various genes. This plot summarizes the similarities of samples in terms of their global gene expression level, and for most of the wild type samples to control the clashes together, while most of the genes of non-wild type samples clashes with just each other. So this is a list of genes that I have, but it's only a partial list. And these are the top 5 significant genes that are highlighted. And we were only interested in the 5 genes less than 5% because of how significant the genes were. If it's less than 5%, it's significant. And if it's over 5%, it's not significant. And then this is my R script. As you can see in the top left-hand corner is the codes that I coded to get the data. And at the top left corner at the bottom is different genes. And then if you see CTR_1, that's actually the column names of the control. And for my results, as the top 5 genes, I have the list of the names HT-T-PS. Then you have L2 and XPO5, H2-Q4, H2-BL, and each dot represents a gene. So for this example, the axis indicates the average expression level, and the y-axis indicates the expression difference between the 2 strains, and the red dots at the top are upregulated in the non-count strain, and the blue dots are downregulated in the non-count strain. And for my conclusion, our bioinformatics analysis of RNA sequence data using the RStudio identified 33 genes that have different expression levels in the 2 mice strains. Genes from the major histocompatibility complex, in parentheses, a coding region for surface proteins, class 2 are identified more than expected. RNA sequence with bioinformatics analysis is a powerful approach to help observe the different biological processes between the 2 mice strains. I came to the NSF RU program with zero knowledge and background in information technology, and this internship has taught me to challenge myself and step outside my comfort zone when it comes to the STEM field. And I want to thank my mentors for helping me this summer and Dr. Arnold for this fantastic opportunity.
Thank you so much. So you started the project and you really had to learn R right from R scripting right from the beginning.
Yes, it took me a month to learn.
That's wonderful. And I gather that's less intimidating when you're given you're not doing it in a course, you're doing it for real, and you've got the summer to sort of process all that new direction of. So are you going to, do you think that will have an impact on what you do this fall as far as getting more experience in this area, or?
Yes, so I actually told my mentors that I'm actually interested in probably doing some certifications in IT because it opened my eyes into something that I thought I couldn't do before, but it's actually interesting.
Right. And that amplifies on what Nikita Womack was saying, our guest speaker earlier this week. So that's really great. So I was curious. We have several questions here in the chat, and I'm just going to read Kai Zhang Yi's question. What is your favorite part of this experience? Reading papers, writing R scripts, doing the presentation, or others?
I can honestly say it was the R script for me because when I first came in, and my partner, he was a high schooler and he had IT background and I didn't. I was so behind and I thought I really couldn't do it. But they told me just take my time. R is hard to learn, but you can actually do it. And when I took the time to learn the packages and when we did our little meetings and I asked for more help, I was like, okay, I can actually do this. I didn't think I could. And it's a lot that actually go into it.
So Tanya Rosario has a question for you. What was the phenotype of the knockout strain? What did it affect? How did it affect the microglia? What did it do?
That's actually a good question. And to my knowledge, I don't think it really affected anything. It's just different levels to all the different allergy genes.
Okay. And you had you highlighted the 5 genes, including MHC, which was affected, but you were really, the candidate list was much, much larger, right?
Yes, it was like 1,700 genes, I believe.
Okay.
It was a lot.
Right. So you went for the needle in the haystack and found 5 candidate genes. And here's a question from another member of the audience. What was the biggest lessons you learned this summer?
The biggest lesson I learned is to have confidence in my work and not doubt myself and understand that I can do anything I put my mind to.
Oh, that's wonderful. Yeah, I just wish there were more of these programs, because even with Sally O'Connor's, all her efforts over the years, there are only about 100, and I may be overestimating, there are only about 150 of these programs in the biological sciences, and the typical size of those programs is about 10 students, like here. And so that's like only 1,500 students that each year get reached like you. So yeah, we'll hope that she keeps pushing for more so that we can have more people that have this opportunity.
Yes,
So,
definitely.
yeah. And so in that, I really don't know much about mice. I work on fungi. I'm sort of curious what even a microglia is. It sounds interesting. Is that, where does that, what is that thing?
So from the papers that I read and from having talked to my mentors, because I didn't know what it was either. It was very like, oh, I never heard of this scientific word before. It's like a type of cell. That's the best way I can put it.
Okay. I've got another question from the audience. You have inspired many from your community. What advice would you give someone who wants to do what you want to do? How do you succeed?
Advice I would give, because I know a lot of like teenagers and undergraduate college students, they think being in the STEM field is very hard, but I feel like we don't have the knowledge to opportunities that's out there in the STEM field. You don't have to always go to medical school or always do research. It's other things that tie into that gap. Mm-hmm. So always work towards that and try to get as much knowledge as you can.
Oh, that's wonderful. So I think what we'll do is now give the speakers a break, and we'll be back at 10. And thank you so much, Ayana. We'll be back at 10:15. There'll be I think probably a countdown will be on the screen, and you can join us again at 10:15. Thank you for joining us. Okay. Great, that was awesome. That went very well. Yeah, that was one.
The bathroom or anything?
No, I'm fine. I'm sure Yinping was calling for a reason,
Okay.
and I will just call her back. Hi, I was being broadcast, so I couldn't answer. we're now going to hear from Cameron Felt. She's a rising junior at the University of Georgia in the genetics department. And she has been working on a fundamental symbiosis between almost all land plants and fungi. And this symbiosis dates back about 450 million years. And it's an example of how we can involve undergraduates in the cutting edge of discovery on these very basic biological questions. And in this case, her work is not only funded by the National Science Foundation, but also by the Department of Energy, which has put her into places that you probably didn't really imagine she'd be involved over the summer. So with great pleasure, I turn it over to Cameron.
Hi, everybody. Thank you so much, Dr. Arnold, for that introduction. Just gonna share my screen. Okay, like Dr. Arnold said, my name is Cameron Felt, and I did my summer project on studying AMF colonization and root morphology under varying nitrogen, phosphorus, and potassium ratios in sorghum bicolor. And so I just want to start off with some background. So what is AMF? So AMF stands for arbuscular mycorrhizal fungi, and it is a fungi found in soil that has a symbiotic relationship with plants, providing vital nutrients to the plant, such as nitrogen and phosphorus, and protecting the plant from fungal pathogens. In return, the plant provides lipids and sugars to the fungus. And this AMF symbiosis can be traced back 400 million years ago, and this mutualistic relationship aids in the development and growth of plants. And 70 to 90% of plant species can form these interdependent relationships with AMF. So just some more background. So how is AMF different than other fungi? So AMF is an endosymbiote rather than an ectosymbiote, meaning that this fungus lives inside the plant that it is helping. And so you can see in this first image here on the right side represents an endosymbiote while the left side represents an ectosymbiote. So just visually you can get that representation. And also AMF can provide plant nutrition, improved soil quality, and plant growth. And so this next image on the right shows the AMF structure microscopically. So the AMF colonizes the roots of plants and forms vesicles, arbuscules, and hyphae and the roots. And so these blue lines that you see are the hyphae that connect the AMF, and the blue structures inside the roots are the arbuscules. So AMF and sorghum bicolor. So sorghum is the world's fifth most important cereal after maize, rice, wheat, and barley. And in the event of climate change and global warming, sorghum could be a viable solution to farmers due to the fact that it requires less water and can thrive in varying climate changes. And sorghum has been recognized for many years for its relationship with AMF, varying among different genotypes. And due to its high productivity and stress tolerance, sorghum was a great plant for this experiment. So overall, our project goal, so like I mentioned, AMF is known to aid plants by colonize with nutrient uptake, and sorghum is known to be colonized by AMF, but it's unknown how fertilizers with varying nitrogen and phosphorus ratios will affect the AMF colonization and morphology of the plant roots. Therefore, we chose 3 sessions of sorghum and treated them with different fertilizers for 4 weeks under greenhouse conditions. So we're just gonna walk y'all through the methods and how we really did this experiment. So the seeds were planted in a greenhouse in garden soil to allow the seeds to germinate. And they grew for about 2 weeks and then were transferred into their individual pots. And a total of 24 plants were used for the entire experiment, and 8 plants were used per each accession. And each accession had 3 different treatment groups. So we had 2 controls, 3 plants for treatment A, and 3 plants for treatment B. And each plant was individually potted in a pot that was 1 cubic feet. And the soil that we decided to use was a 1-to-4 ratio of Wellbrook soil, which is a local soil here in Athens, Georgia, to turfay, which is a greenhouse soil that is autoclaved and is used to improve soil quality. And the 3 sessions that we used, the PI numbers were 152963, 330182, and 562985. So this picture here on the right shows our setup in the greenhouse once all the plants were planted. And this picture was taken 2 weeks into the treatment. So the plants were already pretty big. And the treatment plan that we used, so treatment A consisted of a 10-30-20 ratio, and treatment B was a 20-10-20 ratio. And these plants were treated twice a week for 4 weeks, and half a tablespoon per gallon was used for the rate of fertilization for the plants. And again, this is just an up close picture, so y'all can see what the plants looked like. And so the first set of data we were able to obtain while harvesting these plants. So we harvested the roots, but we also harvested the canopies at the same time. And we placed them in bags and into a drying oven at about 118 degrees Fahrenheit. And we left them in there for a few days. And we took the dry weights of each sample and averaged them. And that's what you see here on this graph, an average dry weights of the sorghum. And on the y-axis, we have dry weight in grams, and the x-axis, we have the treatment groups. And each color represents a different accession. So as you can see, treatment group B tended to have the highest dry weight. And this agrees with the fact that farmers typically use higher nitrogen fertilizers to produce a higher biomass in plants. And our treatment group B used the highest nitrogen levels. And so continuing, the next set of data that we were able to obtain was from the roots. So we imaged the roots and ran the images through the DIRT software, and DIRT gives you a lot of different data. So I just picked a few to analyze. And so first we have area. So each of these graphs represents a different accession, and the y-axis is the area, and each box, different colors represent different treatment groups. So blue, we have treatment A, orange, we have treatment B, and gray is our control group. And so for the area across the accessions, they were pretty similar. And as you can see in the graph, the boxes overlap. So we came to the conclusion that the fertilizers didn't affect the root morphology in regards to area. And the next data that we analyzed was stem diameter. Again, we have the accessions for each graph, and the y-axis represents the stem diameter, and each color of the box represents the same treatment group as the last graph did. And overall, the control groups tended to have a smaller stem diameter, while the fertilized treated groups had a larger stem diameter. So we could see here that different nitrogen and phosphorus ratios did affect the phenotype of stem diameter in this experiment. And so other results that we are currently working on, when we took the dry weights, we also took leaf samples and sent them off to a team in Arizona, and they are measuring the nitrogen and phosphorus levels in each of these plants. So we're just waiting for that data to get back to us. And the next step in this project would be to stain the root systems and image them microscopically in order to determine the AMF colonizations among all the different accessions and treatment groups. I would also just like to add that the DIRT software gave me a lot of data, and I'm still working to work through all of that data to determine which phenotypes are significant in this experiment, and running different statistical tests as well. So yes, that is what I have. I will take questions.
Wonderful. So what was the most challenging thing you had to do this summer?
Well, this summer as a whole, I would say for my project, the DIRT software was probably the most challenging, just because it gives you so much data that you kind of just don't know where to start. So just figuring out what was significant, and I'm still working on doing all of that. So I'm excited to continue with that part. So yeah.
Wonderful. So it's like, and had you, like Ayana, have you had prior bioinformatics experience before this?
I have had no bioinformatics experiments, so that was definitely a challenge for me. But it was very fun to learn. I learned a lot of new things this summer throughout this project. So.
Right. So Noah Legal has a question for you. He's a bioinformatics graduate student here at
Okay.
Georgia. Great work, Cameron. Statistically, do you know of a way to discern how significantly different the root sizes would be between the treatments?
Yes. So great question. So I am going to do an ANOVA test, like a one-way test on all of this data that I have presented and the data that I have. So that is one way that I'll be able to determine how significantly different they are with area, root sizes, stem diameter, all of that good stuff.
Wonderful. That's great. And let's see, so having had this experience, did it have any effect on sort of, what you felt like you needed at Georgia in the fall, or, in your coming year here?
Yes, I am definitely taking more statistic classes this coming year in order to better my understanding of data analysis and the results I get and things like that. So yes, it has definitely affected that.
Right. Yes, because there's nothing better to actually have a real problem in hand like you do. You have real data, and then use that as a motivation for acquiring that new hard skill, which it would represent. It'll be a skill that will help you in whatever path you take. So that's great. Could you say a little bit more about, what is the process by which you actually visualize, because you've done a lot of this, you visualize the fungi in the roots. Can you just tell people what that involves?
Yes. So a lot of this summer I did annotating on microscopic images of the AMF. And so that entails like the staining of the roots and imaging them and then annotating them and then running those images through softwares that will be able to tell me the percent AMF colonization in those roots as well.
Okay.
So that's something I'm hoping to be able to accomplish in the future with this project.
Okay. And does the plant have anything to say about this, about getting colonized? Or is it totally up to the fungus?
Yes, it's definitely the plants as well. Kind of back and forth with nutrients. It definitely varies. That's partly what I'm really interested to see with these varying nitrogen to phosphorus ratios, how that affects the plant's relationship with the fungus and the fungus relationship with the plant. So
Right. Wonderful. So I think what I will do now is thank you for this wonderful project. Thank you for all your work in the field. What she hasn't told you is that there is a lot of
thank you.
it's my pleasure to introduce Christina Juritich. She came to us from Truman State University. She has a little bit different background. She's actually has a strong interest in mathematical biology. And so in some ways, this is really giving her an opportunity to put feet to that interest and get involved in problems in bioinformatics and mathematical biology. So she chose as her mentor Dr. Duda Bensison, who's in the Institute of Bioinformatics. And she is going to be telling us about a project to look at aneuploidy in natural isolates of Saccharomyces. Hopefully I got that all right. And again, she'll have a focus on the bioinformatics aspects of studying this. Please, I'm turning it over to Christina.
Thank you, Dr. Arnold. Yes, so as you said, my name is Christina Jurtich, and this summer I was looking at the frequency of aneuploidy in the wild yeast Saccharomyces paradoxus. So just to start off by kind of describing what aneuploidy is and why it's important to study. Aneuploidy is the gain or loss of one or more chromosomes in a cell. So yeast specifically have 16 chromosomes, and paradoxus is found as diploid in the environment, so it usually has 2 copies of each chromosome, shown here in the figure on the left. And so aneuploidy can occur whenever a cell is undergoing reproduction and the chromosomes are segregating into 2 offspring cells. And so if the chromosomes segregate improperly, then you're going to end up with too many or too few chromosomes in the resulting cells. So on this figure on the right-hand side shows where this chromosome has gained a copy of chromosome, or this cell has gained a copy of chromosome 9. So when this happens, the extra chromosome actually results in kind of an overamplification and overexpression of all the genes that are contained on that chromosome. So the organism might experience effects such as like excessive growth or kind of upregulation of metabolic processes in the cell. And then the opposite effect that can happen is that you can lose a chromosome. So you might only just have 1 copy of a chromosome in a cell, shown here by just 1 copy of chromosome 1 as compared to 2. And so in this case, you might see the opposite effect happening, where you're losing those genes that were contained in that chromosome. So you're going to have maybe stunted growth or underregulation of different processes in the cell. So based on like whenever you gain or lose a chromosome, it creates an imbalance in the genomes, which can be typically pretty detrimental for the organism. So we're just looking at this in yeast because it is a relatively easy organism to study and there's lots of data availability. But then the goal would be to kind of gain an understanding of how aneuploidy, like the frequencies and the effects that go into how the organisms actually tolerate aneuploidy, and then apply it to other areas where we see aneuploidy, such as like is in human disease or human health. So my project this summer was to just look at the rate of aneuploidy in Saccharomyces paradoxus, and then compare it to the rate of aneuploidy in its closest relative, Saccharomyces cerevisiae, which was just recently looked at. So paradoxus is a completely non-domesticated yeast. It's found in the wild, so it's usually isolated from trees, oak trees specifically, but it can also be found on other plants like flowers or sometimes on insects and fruit. And then its relative, cerevisiae, is a bit of a more diverse species, so it has what we call different clades, so it can be found non-domesticated, so on trees or fruit, but it's more commonly known for being used in domesticated settings, so it's commonly known as baker's yeast, and it's also used in the brewing industry. So what this study found was that the frequency of aneuploidy differed in cerevisiae among the clades, so it was less common in the wild clades and much more common for the cells to be aneuploid in the domesticated clades, so the strains used in baking or in beer making. So the point of my project was to kind of see how paradoxus would compare to the aneuploidy frequencies that we saw in the wild clades of Cerevisiae, because as Paradoxus is a non-domesticated species, we were hypothesizing that it would be very similar to the rates seen in the non-domesticated Cerevisiae clades, which we also called the ancestral aneuploidy rate, so whenever before domestication. So this is the pipeline that I followed for actually looking for the aneuploidies. So I started off by collecting the data. So I just took whole genome sequences that had been sequenced by other researchers and published in other studies and downloaded them to the software. And these sequences were all created using a method called Illumina sequencing. So they were sequenced in very short segmented reads. So in that, the whole sequence isn't one long strand, it's in multiple different parts. So then the point of the mapping step is to take these small parts of each strand and compare it to a reference sequence that we have for Saccharomyces paradoxus to create one long sequence for the strain. So this is an example of a strain that we were mapping to the reference. And it's kind of like analogous to having a bunch of different puzzle pieces and you're comparing it to the picture on the box to put it all together into one cohesive unit. So then from there, we create the consensus strand, which is just kind of gluing all those pieces together into one long sequence, and then also comparing the strain to the reference, going base by base and picking out the bases that don't match up, which are called the alleles in the strain that don't match up perfectly to the alleles in the reference. Those are called SNPs, so single nucleotide polymorphisms. And then from there, we can create these lovely plots, which are what I was able to read and use to actually call aneuploidies in these cells. So this is a blow-up of those two plots from before. So this top plot is called a B allele frequency plot. And this bottom one is the read depth. So sorry if it's a little bit small on the screen. We can try and zoom in a little bit. So these two graphs represent just one chromosome from one strain that I looked at. And each point on these dots, these little black dots, represents a point where this strain was different than the reference. So those SNPs that I was talking about earlier. And along the x-axis, you can see it's the genomic position. So it kind of records like where the SNP was seen in the genome. And then on the y-axis up here, you have the proportion of base calls that differ from the reference. So this kind of shows you the whether a strain is homozygous or heterozygous. So since you can see a lot of these points up here kind of at the 1.0 mark, that would indicate that it's homozygous, since in a homozygous strain, the cell would have two copies of like the same allele, so it's always going to be like the same. It's always going to be different than the reference. But in a heterozygous strain, you might see a collection of points at the 0.5 mark as well, because in a heterozygous strain, you have two different copies of the allele. So half the time, one chromosome might have the same copy as the reference, but the other chromosome might have a different copy. So therefore, the proportion is that 50% of the time it's different than the reference. So then down here at the bottom, each point down here corresponds to a point in the top plot. And this just graphs the read depth of this particular chromosome in this strain. And the read depth is just something that's kind of generated based on the sequencing and mapping steps. But what we're most interested in is what the mean read depth is for each chromosome. So this black line here shows the mean read depth. And for this strain, for this chromosome, it's 50. And so what we would want, what we would want to see is that every single chromosome in this strain has an approximate read depth of 50. And what we would notice if it was an aneuploid or not would be if the read depth for one chromosome was significantly higher, say a mean of 100, or significantly lower. So to make that easier to see, I made excuse me, a graph that looked like this, which is just a sort of compilation of all the read depths from one strain. So this has 16 columns representing each chromosome in this strain WX15. And so this is just kind of like turning the bottom plot from the previous slide switching the orientation and putting all the chromosomes in a line. And these blue lines right here represent the mean read depth that was shown before with a large black line on the slide. So typically all of these lines would line up kind of like in more or less of a straight row. But occasionally if the strain is aneuploid, you would see in this case that one chromosome's read depth is significantly far out of line from the rest. So this looks like an aneuploid, and then I would have to do some calculations to actually verify kind of what kind of aneuploidy it is. So you calculate what's called the read depth variance, and you just take the outlier read depth, so whatever this value is right here, which is shown at the top, is 149. And then I would calculate the median read depth out of all 16 chromosomes, which for this example was 101. And so that means that for this particular example, chromosome 2 had a read depth 1.5 times greater than the rest of the chromosomes. So it was 1.5 times bigger than all the other chromosomes in this strain. So based on these parameters that I was using, it shows that chromosome 2 actually seems to be a trisomy. So it has 3 copies of chromosome 2 rather than 2 copies like normal. And the other options would be if a variance was less than 0.5, you would have lost a chromosome, as I showed before. And then if there's a variance that's greater than 1.75, it's relatively uncommon. That would mean that you had gained 2 or more of extra copies of that chromosome in that particular strain. So what did I find overall? I ended up looking at a total of 238 paradoxus strains that were available, kind of after applying some filters and removing some strains that were pretty low quality and difficult to create graphs for. And I actually found that there was only 7 aneuploidies out of all those 238 strains, which is a frequency of 2.94%. So the rate that we were comparing it to in Cerevisiae for the non-domesticated strains was approximately 9%. And so whenever I compared these two values using a binomial test, I found that these two were actually significantly different from each other. So the rate of aneuploidy in Paradoxus was even lower than the rate of aneuploidy in non-domesticated strains of Cerevisiae, which is surprising because it was actually even lower than we were expecting it to be. So this little table down here kind of shows just a breakdown of the types of chromosomes that I saw that were aneuploid, or which chromosomes were. And so as I kind of said earlier, when describing aneuploidy, whenever you amplify or lose a chromosome, you're also you're amplifying or losing the genes that are contained on that chromosome. So the theory is that chromosomes that have a lower gene content are more likely to be tolerated when aneuploid in the organism. So for example, chromosome 1 in Paradoxus only has 90 genes. It's very small and very low gene content. It's the lowest out of all 16 chromosomes. And so that kind of makes sense that I found chromosome 1 to be aneuploid in 4 separate strains. Chromosome 9 similarly is pretty low in gene content. It has 205 genes, and it's among the bottom 5 for gene content of all the chromosomes in Paradoxus. And I only found that amplified in 1 strain. And then chromosome 2 is actually surprising to see aneuploid a little bit because it does have a relatively high gene content. It's among the top 4 or 5 for the most, containing the most genes out of all the chromosomes in Paradoxus. So these are just kind of my conclusions in a nutshell. I found that Saccharomyces paradoxus was very rarely aneuploid, the rate of 3% as compared to the rate of 9% in the non-domesticated Cerevisiae strains. And I also found that it's almost always homozygous. So that was based on those B allele frequency plots seen before. And this is useful for knowing whenever you're sequencing strains. That way it's easier. You kind of know that it's easier to sequence Paradoxus. As it is homozygous, you don't need to worry about creating a monosporic derivative to make it homozygous, to make it easier to sequence. And then lastly, I found that the chromosomes that are most frequently aneuploid do tend to have a lower gene content. So all of this to say that it kind of does confirm what we were looking for initially. We did see that the rate of aneuploidy was very low in this non-domesticated species. And it was surprising to see that it was actually even lower than we thought initially. So it kind of shows that perhaps those strains that were seen to be extremely aneuploid in Saccharomyces cerevisiae, the relative, those strains are domesticated, so potentially the human influence has a major effect on why those strains are showing aneuploidy, because obviously in their natural state of non-domestication, they are very rarely aneuploid.
Thank
So,
you.
yeah, just wanted to thank Dr. Bensassen for being a wonderful mentor this summer, and then Eduardo Scopel, one of the lab members, for also being a second mentor and helping me along every step of this project and answering all my questions. And then their whole lab just for being very welcoming and great to work with this summer. And then, of course, Dr. Arnold for running this wonderful RU this summer. Thank you.
Thank
That's
you
all.
for this wonderful talk. And I just had, one or two questions. Do you, so it's interesting that Paradoxus has, a lower aneuploidy rate. So do you know if it's, whether it outcrosses, whether it engages in mating less often or something like that than its domesticated cousin?
I'm not entirely sure. That's a good question. Yeah.
Yeah, because it is striking. I'm also curious, the other related question, what is the aneuploidy rate in us?
It's pretty low. I don't really have a number for that, but in
Okay.
humans, aneuploidy is not well tolerated at all. So like in humans,
Right.
there's only 5 chromosomes that can actually be aneuploid in a cell and result in a viable offspring. So like out of the 23 chromosomes that we have, only 5 of them actually result in
Right.
viable offspring. But even then, that's really detrimental effects to the organism. Like there's phenotypic disabilities, kind of sometimes mental capabilities altered.
Right.
Yeah, so it's not very well tolerated, which is kind of one reason
Right.
of why we're looking at
So,
how it works.
Dr. Kaijiang Yi had a question. Have you compared the gene contents of the 3 aneuploid chromosomes to other chromosomes? And,
Yeah.
Yeah.
So I didn't put like the total kind of like numbers for each chromosome, but chromosome 1 does have the lowest gene content out of all 16 chromosomes with 90. And then kind of like on the bottom like section of all 16 chromosomes, chromosomes 1, 3, 6, and 9 do have the lowest content. So those 2 I did see amplified. But chromosome 2 actually has a relatively high content compared to the other chromosomes in the yeast cell.
Okay. And Tanya had a question, and I'll probably have to call it after this one. Do you have any ideas why wild strains would be less tolerant to aneuploidy than domestic ones?
So yeah, one thing that we kind of that was like, I guess, seen, this was more seen in Cerevisiae. I didn't really look at too much like of the actual tolerance of aneuploidy and Paradoxus, just more of like the numbers of it. But in Cerevisiae, the kind of thought was that aneuploidy is somehow helping these cells that have been domesticated, so like helping them survive in their environment. So it's not really necessary for the cells in nature really to evolve aneuploidy or be aneuploid. So I'm not really sure like what kind of factors are affecting why they might be less tolerant, but the idea is that they might be helping the other organisms in domesticated situations undergo stress.
Thank you so much. Thank you for the wonderful talk. And
Thank you.
I'm grateful that you were in the program. And we will now move to our next speaker, Janebi Benoit. Here he is. He's from Bunker Hill Community College in Boston, right? Or is it Cambridge? It's probably
in Boston.
It is in Boston. And he worked with Dr. Justin Bale on the efficacy of vaccines, which are of all of interest to us right now. And Dr. Bale is also one of the co-PIs on our COVID-19 group. So I'm looking forward to, what you can tell us about good vaccines. Please proceed, Janebi.
Sure. Thank you, Dr. Arnold. So let me just start, with my introductory slide. As Dr. Arnold mentioned, I'll be presenting today about the phylogenetic distance as a measure of vaccine efficacy. And just a brief outline of my presentation. I'll be speaking about influenza and specifically the influenza A H3N2 subtype, which is what we researched this summer. And to explain that further, I'm going to speak about phylogenetics, then my methods of research, and my results and some next steps. So to start, for a brief overview of our project, what is seasonal influenza? It's important to know that seasonal influenza is made up of 4 main subtypes, which is the H3N2, the H1N1 pandemic influenza, then we have influenza B Victoria and B Yamagata. The influenza virus is from the Orthomyxoviridae family of viruses, and those viruses have a negative sense single-stranded RNA segmented genome. And as you can see on the right-hand side of my slide is a very nice diagram of the influenza virus. And if you pay attention to those orange and blue surface proteins, we have those 2 main surface proteins on the virus, which is the HA, the hemagglutinin, and the NA, neuraminidase proteins. And those are what allow the virus to enter and exit a host cell. So let's look at why influenza is important. And this chart is from the CDC morbidity and mortality, well, for influenza, the influenza burden, disease burden for the virus. And each year from 2010 to 2020, there have been at least 20,000 deaths that have been influenza related. So that's a lot of deaths, and that's significant morbidity and mortality, and that's why we look at and study influenza. And now for our reasons for studying H3N2 subtype. So what we did was look at the CDC's morbidity and mortality weekly report, which is a report the CDC publishes every single week of each year. And typically, H3N2 is the most prevalent influenza subtype among seasons. So that stood out to us, and we decided to look further into it. And H3N2 is constantly changing due to antigenic drift, which simply put is just the gradual increase in genetic variation of viruses due to mutations. And these mutations can change the surface proteins that I talked about earlier that the host immune system recognizes to eliminate the virus. And these changes can cause the virus to escape immune pressure. So because of antigenic drift, it is difficult to create a vaccine that is highly effective for a long period of time. So forecasting for vaccine epitopes is a major goal of researchers to help create effective vaccines to stop epidemics, and also to possibly even come up with a method for finding universal vaccines. So in my study, actually, we aimed to find, or we studied how genetic distance from a chosen vaccine candidate could show vaccine efficacy. And we used mainly what's called patristic distance metric, calculated from phylogenetic trees, as seen below in this slide, to compare circulating lineages against the vaccine strain for a particular season. And I'll talk more about this further along this talk. So what is phylogenetics? In this study, we used phylogenetics as just a tool to understand the distance between circulating strains and the vaccine strains for an epidemic season. The epidemic season we looked at for influenza H3N2 was the 2016 to 17 season. And phylogenetics uses sequence data and other information to create a true representation of relatedness between different taxa, or taxa meaning just samples, like samples from the host, and the host being humans in this case. So the trees below are just simple trees. The one on the left is a very general tree so that you can understand if you're unfamiliar. And the tree on the right is actually the tree from next strain of the novel coronavirus pandemic. So as I mentioned earlier, trees can help describe relationships among taxa, and they allow for several other measurements to be extracted. And that, if you see, I have highlighted in red patristic distance, which is what I'm going to expound on right now. Or before I expound on that, I'll speak about my materials and methods. So first, we started off by using the NCBI's database to get the HA sequences of the H3N2 virus. And we also used the vaccine strain from the World Health Organization, and that was collected from GISAID, which is also another database. We then performed multiple sequence alignments using MAFT software, and we then visually inspected the alignment in a software called AliView. This was done to remove gaps from the nucleotide sequences and align the start and stop codons of the sequences properly to allow for comparison of sites across the sequence. And in order to understand the relationship of circulating viruses and the vaccine, we then partitioned the season into 5 successive time points. And we created sequence data sets for each time point, adding sequences from the previous time points to the successive time point. The vaccine sequence was included in each data set as well, just to show the genetic distance of the vaccine to the strain circulating in a particular time point. We also used a software called IQ-Tree to perform maximum likelihood phylogenetic reconstruction of sequence data sets for each time point. And finally, which was one of the main parts of the project, was the patristic distance matrices. Now, these distances that I mentioned are outputted in the form of matrices, very, very large matrices. And actually, for my project, the H3N2 data collected from NCBI was like 2,700 sequences. So it was a lot to deal with. And the trees I'm going to show you later on would depict that. So we used patristic to calculate those distance matrices, and the resulting phylogenies to create, the resulting phylogenies created the distance matrices. And then we used, my lab partner used multidimensional scaling so we can get a nice figure that I'm about to show you. So these are the phylogenetic trees that I just spoke about from the H3N2 virus, and the successive time points, the 5 successive time points that I spoke about. As you can see, the trees get more and more thicker as the time goes on. And here we have the multidimensional scale of patristic distances. So we can see that over time, the vaccine candidate for the season, denoted by the purple triangle here in the middle, it goes from being closely related to the taxa present to gradually being farther away from the major clusters that form. So if you look at T1, this depicts the first phylogenetic tree that you saw, and then T2, and so on and so forth. You can see the clusters getting larger and deviating. This also represents the antigenic shift that we spoke about earlier. So just to move on, a further discussion of my project. From the figures that I just showed you, we determined that antigenic drift, is very evident in our study. And the antigenic drift from the vaccine candidate was close to all major clusters present. And this might be partly why in the next season, vaccine candidate for this season was also used. It is important to note that this was a very active season, and other changes in the environment and the virus biology might also be important. So therefore, an important next step is to do this analysis using another surface protein, the neuraminidase, and also to expand the analysis to multiple seasons so that we can come to a conclusion about this research. That'll be all for my presentation, and I'd like to just give a few shout outs and acknowledgements. I'd like to thank Dr. Arnold with the University of Georgia for accepting me into this program, and I'd like to also thank the NSF, along with my lab PI, Dr. Justin Ball, and the grad student that I worked with for most of my time here, who has been very, very helpful, Lambo. Thank you, Lambo. I'd also like to thank Gianni for going through some of the submission scripts that I used to submit work onto the computing cluster. And I'd like to thank Dr. David Campbell, who was my research mentor from my last REU, that gave me my introduction to research, and my high school teacher, Miss Wilson, for those excellent recommendation letters. Thank you.
Thank you for this wonderful talk. It's fantastic. So, I gather you, hopefully you'll continue to look at the other surface proteins, and career-wise, you're pretty directed towards doing bioinformatics. Would that be a good assessment?
It would. So my major is actually electrical engineering. So that's probably,
Okay.
I'm probably the rarest major here. But I've always been interested in biology, and I always wanted a career in biology, and hence my reason for doing this research this summer.
And so the data that you got, it was from, so you got it from the CDC. The sequences came from GSAID, but you had also the incidence across years, which I assume was the data that you got from the CDC. Do you think you get a very different story this year with everybody wearing masks? Was 2020 in that picture that you showed in your slides?
So we didn't study the 2020 season, but we
Yeah.
did look at it briefly to choose which season we studied. And
Uh-huh.
the mask wearing in 2020 definitely helped because, so we used actually the CDC's MMWR reports to decide, and there were barely any influenza cases for 2020. So 2016 to 2017 was actually the most popular season in the past 5 years.
Okay, that's good news for us. So I'm going to end with one question from Noah. Awesome work, Janebi. Good to see your hard work from the summer. I wanted to ask what metrics you could collect to learn how well supported your trees were. So I think he's asking about bootstrapping and things like that.
That's a great question. So we only use the maximum likelihood framework for this. Eventually, we were also considering carrying out this in a Bayesian environment and also performing ancestral sequence reconstruction.
Right.
We haven't done that yet, but those are some possible steps moving forward.
That's wonderful. Thank you so much, Janevi. We're grateful and privileged to have worked with you. And I'm now going to introduce our next speaker. She's Yivrin Nguenta Nkecha, and she's from Georgia State University. She's not far away, but I'm curious, where you're actually residing. It's probably could be far away. We don't. And she had the opportunity to work with Dr. Travis Glenn. He's the director of the Institute of Bioinformatics. And he is also has a focus on infectious diseases. But Yivrin is going to take us in a different direction. She's going to look at microbiomes and how that tells us something about being infected by the Lone Star tick, which I gather none of us want to get bitten by. So on that note, I'm going to turn it over to Yivrin, and we look forward to this wonderful talk.
Thank you, Dr. Arnold. So, I had the pleasure of working with Dr. Glenn's lab this summer, and our project was examining possible variables that influence microbial diversity of Lone Star ticks. So, why study tick microbiomes? Ticks are biological vectors, which are organisms such as ticks, fleas, mosquitoes, or snails that feed on the blood of hosts and can transmit pathogens between these hosts. And pathogens are usually distributed by contact with the blood of the organism or from being bit. Studying tick microbiomes and how pathogens interact with other microorganisms can give us a better understanding of these pathogens, which can in turn help us control their spread and effect on human populations. So in this picture here, I have 3 different types of ticks, which are the blackleg tick, dog tick, and the Lone Star tick, which is the tick that we will be looking at today. So Amblyomma americanum, which is also known as the Lone Star tick. So I have a picture here of Lone Star ticks at different life stages. Here is a larva, a nymph, adult male, and adult female. And the adult females are easily identified by a white spot on their backs. And you'll usually find these ticks on the eastern half of the United States. So diseases. So rickettsia are different types of parasitic bacteria with the genus Rickettsia and order Rickettsialis that are gram-negative and habituate within the cells of the host. And in this picture here, you're able to see red-stained rickettsia in the cells of a tick. The most common disease contracted from Amblyomma americanum is ehrlichiosis, and it's caused by the pathogens Ehrlichia chiefensis and Ehrlichia ewingi. And some symptoms of these diseases include fever, headache, chills, nausea, muscle aches. And specifically with Ehrlichia chiefensis, you get rashes. And these symptoms are what you commonly find with tick-borne diseases, and you can classify them as flu-like symptoms. So a lot of people call tick-borne diseases the summer flu. More diseases you find from Lone Star ticks are the southern tick-associated rash illness, tularemia, and Heartland virus. So 16S rRNA amplicon sequencing. So the 16S rRNA is a target gene composed of 9 variable regions that we can use to identify bacterial species and taxonomic composition of microbial communities. So in this picture here, you see these 9 variable regions which differ between species, and these blue areas are conserved regions that are the same between species, and we're able to detect these regions and use primers to amplify any targeted region that we want to look at. So tick collection, identification, and DNA extraction. So this was performed by the Southeastern Cooperative Wildlife Disease Study, and 530 ticks were collected from 3 different habitat types in Piedmont, Georgia, in the Piedmont region of Georgia, which are upland, mixed hardwood, pine, and bottomland hardwood. And on each site, they had 25 meter transects, and tick drags were performed for 500 meters at each site along the transects. And in this picture here, I have a woman performing a tick drag, which is her dragging a piece of fabric that allows ticks to easily hatch on, and then you turn it over and you're able to collect the ticks. And we're able to identify the ticks by species, sex, and life stage using published dichotomous keys, and DNA was extracted using QIAGEN, DNC blood, and tissue kits, which contain spin columns, buffers, proteinus K, and collection tubes. Library preparation, sequencing, and demultiplexing. We prepared 16S amplicon libraries in order to have a better understanding of the tick microbiome. We prepared these libraries by amplifying the V3 and the V4 region. And we use that using the 341 forward and the 785 reverse primers developed by Kenworth and other scientists. And we're able to do this using a Kappa Hi-Fi hot start kit. During library construction, the added barcode sequences in order to, and we added barcode sequences in order to index the samples. Following indexing and final cleanup of the library products, we sequenced those on an Illumina MySeq 300, PE300, and after sequencing the libraries, we were able to demultiplex using the software Mr. Demuxy. So what is Chime 2? So Chime is the industrial standard software for illustrating microbiomes, and it stands for Quantitative Insights into Microbial Ecology. And we're able to analyze these microbiomes from amplicon libraries targeting the 16S rRNA gene. And the figures produced by Chime give us a visualization of the operational taxonomic units and their abundances. And operational taxonomic units represent groups of related organisms. We're able to use Chime 2 in a program called Jupyter Notebook to analyze our sequencing data from this project. So Chime 2 full 16S analysis workflow. So with our raw sequences, we're able to demultiplex those and visualize those sequences. And we also denoised and dereplicated them, which is basically getting rid of any low quality reads or any chimeric reads and combining any reads that were too similar. And we're able to also train our taxonomic classifier. And from that, we could classify our OTUs and produce our figures. So the first graph that we made was an alpha rarefaction graph, and on the y-axis you can see observed OTUs, and there's only 35, so these microbiomes aren't that diverse. And on the x-axis you can see sequencing depth, and all these different colors are all our different samples, and what we're looking for is a plateauing of each sample so that it's no longer increasing, and that tells us that we found all the OTUs in each sample, which lets us know that we sequenced deeply enough. So what pathogens did we find in our tick samples? So all this brown that you see were unassigned in the program, and all the pink that you see here are all that was determined was the kingdom, which is bacteria. But we were able to find a lot of cool pathogens in our samples. Where you see the blue here, it's rickettsia. Where you see this yellow here, it's Coxiella, which causes Q fever. And we were able to find Ehrlichia in 2 different samples, and we also found Borrelia, which is the genus that causes Lyme disease. So alpha diversity. Alpha diversity is a measure where we can compare the diversity of microorganisms within a single sample. And there's 2 types. There's non-phylogenetic, which just lets us know what OTUs are present. And then there's phylogenetic, which lets us know what OTUs are present and their abundances. So our phylogenetic metric was the Shannon index, and our non-phylogenetic metric was observed OTUs. So the Shannon index is basically a measure of diversity and evenness, and the higher the Shannon index, the more diverse a sample was. And by looking at our p-values and looking at all our box and whisker plots where the medians were extremely similar and there's little variation between the ranges in our interquartile ranges, we weren't able to conclude any significant differences between any of our groups. And it was the same case for our observed OTUs. They were all extremely similar, and we weren't able to find any differences. So beta diversity is a measure where we compare the diversity of microorganisms between multiple samples. The phylogenetic metric we used was weighted unifrac, and the non-phylogenetic metric that we used is Bray-Curtis. So what we're looking for is clustering, and I put up here an example of clustering, which is where a group just gathers in one particular area of the plot, and on each axis it's measuring variance. And I put the graphs for life stage, and in neither of these graphs were there any clustering, and it was the same for season and habitat. So conclusion, I was able to identify several pathogenic species such as Borrelia, Ehrlichia, and Rickettsia, and for neither alpha diversity or beta diversity were there any significant findings for life stage, season, or habitat. But I wanted to mention a cool study that I read that was really, really similar to mine, called Factors Affecting the Microbiome of Ixodes scapularis and Amblyomma americanum by Brinkerhoff et al. 2020. So they looked at composition, and they looked at diversity of Ixodes scapularis, which is the black leg tick that I showed you in the beginning, that is the main tick that gives Lyme disease. And they looked at Amblyomma americanum, and they looked at sex, they looked at they looked at sex, they looked at life stage, they looked at environment, and they looked at vertical transmission. And they found between these two species that there are a lot of differences with diversity and composition, but there weren't any differences in regards to environment. But they also found differences with life stage, which I found was interesting. And they also found that adult females had much less diverse microbiomes than the adult males and the nymphs. So this project and others like it lay out the groundwork for future studies on tick microbiomes, which can aid in the advancement of treating tick-borne diseases that affect our health as humans. I'd also like to thank the NSF and Dr. Arnold for giving me this opportunity to do research this summer. And I also want to thank Dr. Glenn, Megan, Marissa, and Mandy for supporting me throughout the summer while working in their lab.
Wonderful talk. That's incredible. So did you have prior experience with bioinformatics before you got here this summer?
No, I didn't. No.
Amazing. So yeah, that's fantastic. You've done this
Thank you.
microbiome analysis. That's pretty amazing. So I'll ask one question while we're waiting for one to come in. One of the nice things about Georgia is we actually have I think an institute at Georgia Southern that is basically studies ticks. And I'm wondering if there are other resources out there that you could tap into as far as having a wider distribution of samples of the ticks. Were your samples all confined to Georgia?
Yeah, all in the Piedmont region.
Right. So here's my first question. It's from Tanya. Did you find any microbes that were not expected to colonize ticks? Any unusual bacteria, for example?
I did, but they weren't that much. I think I can, let me go back to share my screen.
Okay. There. Here it comes. That's all right.
Okay, so all of these are different pathogens, but they're all different. So none of them had, we weren't able to see them at high frequencies like we were able to see the ones that I mentioned. So I wasn't able to identify anything too significant.
Right. Okay. So the orange ones are things that are essentially unassigned. They could be very interesting. We just don't know much about them. So it'll be interesting to look at those further. So thank you so much for this wonderful talk. And I'm glad you had this new experience with bioinformatics. And it's wonderful that we could have you as a participant this summer. Thank you very much. So our next speaker is Miguel Iglesias. I think he's originally from Maryland, but he goes to Baylor. And he is also, he's looking at a eukaryotic pathogen. So far, we just heard about prokaryotic pathogens, but he has been working with Rodrigo Di Baptista, who is a postdoc in Jesse Kissinger's lab, and they're working on an important pathogen for people that work on horses. It's called sarcocystis. It has an adverse effect neurologically. So I'm going to turn it over to Miguel, and thank you for what you're about to tell us about.
Thank you, Dr. Arnold. Let me just go ahead and share my screen. So today, I'll be sharing on the impact of annotation manual curation in eukaryotic pathogen genomes. It's a Sarcocystis neurona case study. First off, I'd like to thank Dr. Kissinger and Dr. Baptista, who have helped me a lot this summer and specifically on this project. So to start off with some introductory information, I'm going to go over some definitions. These are going to come up a lot in this presentation, so it's important to really know what they are. So the first is like, what is an assembly? An assembly is when you're discovering the entire sequence of a genome and kind of assembling it into as many contiguous pieces as possible. So an assembly will be the completed sequence of the genome of the parasite we study. What is an annotation? Annotation it's just information that describes the location and structure of a gene. So it'll tell you what the gene structure is and where inside that genome, where in the assembly that is. Manual curation is when you create annotations by hand using available evidence. So a lot of what I did this summer was manual curation, the annotation. And what is Apollo? Apollo is a manual curation software. It's seen here on the right. So we used Apollo to create these annotations so we could learn more about Sarcocystis neurona. But speaking of Sarcocystis neurona, to tell you a bit more about the parasite, S. neurona, it's an apicomplexan parasite. It's best known for causing a disease known as EPM, and it infects horses. It's actually the most commonly diagnosed neurological condition in horses in the country, so it's something that definitely needs to be researched. The genome of S. neurona was sequenced and analyzed in 2015 using Illumina HiSeq and 454 Life Sciences sequencing platforms, so it's been about 6 years since then, and when they did that project, they annotated about 7,100 genes, of which only about 5,900 actually had RNA sequencing evidence. So there's a few genes there that they annotated without evidence, which can create some issues. To talk a bit more about the parasite, the life cycle, it's kind of got, I'm going to talk about two different life cycles. The normal life cycle, it's that you have a sporocyst, so you can see that in this picture. It's eaten by an intermediate host. In this example, that is the cat, not the horse. We'll get to the horse in a second. So then within the intermediate host, the cat, what are known as sarcocysts are formed. They're just a cyst full of the parasite. And eventually the opossum eats that sarcocyst when it eats the flesh of the cat. And then the parasite produces sexually to make more sporocysts, and that completes the life cycle when the cat eats the feces of the opossum that contains the sporocysts. But sometimes instead of an intermediate host like a cat, you have an aberrant host like a horse, which eats a sporocyst. And within the horse, no sarcocysts are formed, so that kind of it cannot complete the full cycle because of that. So instead, what are known as merozoites, which is a particular life stage of the parasite, are in the horse, and they grow into another life stage known as schizons. And then the schizons produce more merozoites, which make more schizons, and it continues to replicate within the horse, and that actually causes the disease EPM. The schizon and merozoite stage are going to be important later, as that is what we actually used for evidence looking at the RNA-seq data within those two stages. So to talk a bit more about the assembly work, in 2015, the old assembly, they used what are known as short read methods. As I mentioned before, the Illumina HiSeq and the 454 Life Sciences platforms. Some of the statistics on that, the N50 value was just over 3 megabases. The largest scaffold was just over 9 megabases. The size was 130 megabases, and the number of mismatches was about 12 million. Mismatches are just unknown regions or unknown nucleotides within the sequence. So there's a lot of mismatches. For some context, the N50 value and the largest scaffold, the bigger the better. So to kind of compare that to the new assembly, where we used, our lab built it with a hybrid method, which involved long and short reads using Oxford Nanopore and Illumina platforms. Some of the statistics, the N50 value is about 6.5 megabases, which is about twice as large as the old assembly. And as I said, the bigger the better. The largest scaffold is also about twice as big at almost 20 megabases. The size of the genome actually decreased a little bit to 127 megabases, and the number of mismatches decreased significantly from 12 million to 265. So to talk a bit more about the differences between the assembly methods, we had to learn more about the difference between short reads and long reads. Short reads which was used in the old assembly, take short sections of the genome and then map them to build the sequence based off of where the reads overlap and figuring out the structure in that way. But what can end up happening is in complex regions and repetitive regions, they'll find these gaps because there's so much of that repetitive information. The reads might be identical because of that repetitive section, so they get kind of stacked over top of each other instead of next to each other. If you kind of imagine like you're building a puzzle and it's got a house, a windmill, and a blue sky with a bird, all that blue sky, when you're building that section, it all looks the same. So if I ask you, where's this bird? You have a short read that represents the bird. Where does it go in the sky? Close to the windmill, close to the house? You might not be able to answer that because all the blue sky is just going to be so similar that you're not going to be able to figure out where in the sky it goes. Versus long reads capture longer sections of sequence. So they're going to include an entire repetitive sequence or an entire complex region so you can actually figure out the structure better. So to go back to that puzzle analogy, imagine you have the bird, the sky, but you also have a piece of the house in that read. You now know where the bird is associated to the house. But you might be wondering then, why did we do a hybrid assembly instead of just using long reads if they're better at determining the structure? And the reasoning is that long reads are not as accurate as short reads. So when you use long reads to build the structure, that you can then use short reads to make sure that the actual assembly you have is as accurate as possible. So which is why we used the hybrid method. So now that we have this assembly, though, why should we re-annotate the genome? Why should we put all this effort into figuring out all the annotation work? The first reason is that in silico gene prediction is not always accurate. So the computer, when a lot of annotation work is done with a computer, it'll predict where in the genome these annotations belong, and it's not always going to be right. You can see that really well in that red rectangle up there. The annotation in the yellow section is something I did, versus the annotations beneath that are computer-generated annotations, and you can see that there's a very large difference between what I did and what the computer predicted. Also, sometimes previous researchers make mistakes. So if a researcher's manually creating the annotation, they might make mistakes as well. And a lot of this happens because they're using old assembly methods that are flawed, or they might just not have enough evidence. If you recall back when I said that there was 7,100 genes annotated, of which only 5,900 have evidence. If you don't have the evidence to annotate, you can't annotate as accurately. And when you improve the assembly, you also you can annotate more accurately. And researchers depend on accurate gene sequences in their research. And if you annotate poorly, you can actually undermine research projects that depend on those sequences that result from the annotation. So now that we have this improvement in assembly and the increase in the amount of evidence, we can actually provide better annotations that are more accurate that can help research projects. So to talk about the design of my actual experiment, our hypothesis was that an increased amount of evidence and a more continuous assembly will provide new insights about the biology of S. neurona. But the real question to answer is, does manually curating the annotation, a new assembly, add enough benefit to the research community? Because it does take a lot of work to annotate the assembly, and though we think that we can do it more accurately, does it provide enough benefit? So what I will do is compare protein sequences from genes found in both the old and new assemblies. So annotations give you those protein sequences. So by comparing the old and new assemblies and the old and newest annotations, we can see how much of a benefit we have provided. So to kind of start with that, some of the methodology. To actually annotate the genome, this is back to Apollo. This is an example of a really easy annotation because you see what the computer has predicted is very similar to the evidence beneath it. You can kind of tell because you can try to match these rectangles to all this evidence. And you can see that the computer prediction was actually very good in this case, which makes annotating the gene very easy. In this case, however, sometimes something interesting happens where a computer predicted 2 genes, and then we find out that it's actually just 1 gene, and you can see that here very well within that red rectangle. So that's something that also happens when you annotate. Sometimes the computer predicts nothing, and based off the evidence that you see beneath that kind of absence of prediction, you can tell that something's going on there. So that's why I annotated a gene there in that yellow section. And so this kind of also shows that when this reannotation effort is going to find things that were missed before. And finally, sometimes you have really good orthology evidence. Toxoplasma gondii is a very similar organism to Sarcocystis neurona. So sometimes when there's a toxoplasma gene within that matches the sequence of the genome, it's represented in with those green rectangles, and we know that what is above matches that because of how similar the genes are between the two organisms, even though there's not as much evidence beneath there. So that's annotating the genome. In this project, I annotated about 200 genes, but now to actually do the comparison, we had to choose which genes to compare. So the gene selection method, in the old annotation done in 2015, about 7,100 genes were annotated, and I selected the 92 with a named family, including those in the ROP, RON, IMC, and SRS families, and those were the ones with sequences as well. And of these proteins, most of them had direct links with pathogenesis that are important to researching this parasite. And the paper done in 2015 also did a lot of analysis on these genes, so they are clearly in of research interest and have been studied previously. So it's important to see if the annotations done previously are correct with these genes. I then used BLAST, which is an alignment software that what it does is if you give it a sequence, a protein sequence, it can find it within the genome assembly. So I gave it all the old sequences and basically told it, can you find them in the new sequence? And what it did, it's similar to when you're looking something up on a web page or in a Word document when you use Control F to look for a word within that document. It's kind of the same way. BLAST does something similar. Here in this left column, you have all these genes that I looked up from the previous paper. I looked them up in the new assembly to see if they were there. And you can see in this right column that it can tell me which contig or which scaffold it is. You can see that they're in different locations. So once I searched up those 92, we're able to find 88 within the new assembly. So 4 were missing, which is pretty interesting actually. But now that we have those 88, we had to annotate some of them because they hadn't been annotated previously. So I used available evidence to actually annotate those genes. And once I've annotated the genes, we have the new protein sequences. So now we have all the old protein sequences and the new protein sequences that match them. So we want to compare the two. So we use something called MAFT, which aligns the old and new sequences. And here in this top image, you see an example of a really good alignment. Those two genes match perfectly. But here on the bottom, you can see that in that top half, they do not align correctly, to show that there is actually a difference between that old sequence and the new sequence. For some further context, by the way, in all of the MAFT things I will show, the top track is the old sequence and the bottom track is the new sequence. And of the 88 genes that we were able to find with BLAST, 83 of them could be aligned. So those will be the focus of my results. And speaking of results, to talk about the results and discuss them further, to start off, of the 83 that we aligned, 28 were completely identical. So between the old and new sequences, there were absolutely no differences. Of the new protein sequences, 39 were longer and 16 were shorter, which I'll talk more about why that's interesting later. Next up, we can talk about why sometimes it gets longer. In this case, you see on the bottom, there's extra information before they start matching. So that'll help make the new sequence longer. And then you can also see that down here, where sometimes there will be extra information after where they'll match for a while, and then the old sequence will kind of stop, and then the new sequence will have more information after that was missed before. So those are two reasons that there's that extra length. Another reason there's extra length is when you have gaps in the old sequence. So you can see that here they match very well, and then there's all those dashes. That's where the old sequence has no information, but the new sequence has a bunch of has new amino acids that were not known before, and then they continue to match again until the end. So 27 of the old sequences had gaps like that. And then 3 of the old genes actually had indeterminate amino acids, which were resolved in the new assembly. If you recall the Old assembly had many more mismatches than the new assembly. It was 12 million versus 265. So that can show up sometime with these X's saying we don't know what amino acid that is in the old sequence, but in the new sequence, we now know those X's are actually in E, W, R, and I. So that happened in 3 of the 83. And then 31 had differences in amino acids not related to length. You can see that here where there's all those, all the dots or times where the letters just don't match up. You can tell that there's a lot of differences between these two sequences, and they're not all related to length. So there were 31 times that happened. So to kind of further explain those results, it's important to understand why this extra length is significant. The first reason is that the new sequence may include a new start codon. So if in the old sequence it started at a certain methionine, and that methionine is present in the new sequence as well, but there's also a methionine earlier that the old sequence didn't find, that's going to add a significant amount of length at the beginning of the protein sequence that was missed before. So you have all this extra information that was missed previously. The opposite can happen with stop codons. If in the old assembly it had a stop codon that's not actually a stop codon, and the new assembly does not describe that as a stop codon, that you'll have all this extra information that happens at the end until the protein sequence reaches the true stop codon that was missed prior. So those two have come from the start and end, where you just miss information in the old sequence. And if we find it, that means we've found important information. All those gaps I was talking about mean that more data was found, and that's found through new exons and coding sequences. You can see that here in that rectangle where there's, in my prediction, there's that green section of the annotation that represents coding sequence, but it's not there in the prediction, but there's clear evidence for it. So when we find new exons like that, you actually create those gaps. To discuss the results of the alignment analysis are promising. It appears as though the new assembly work is more accurate than what was done previously, and 66.3% of the genes changed, showing that what we're doing is important. Things need to be redone because each of those errors can affect an experiment. However, this is limited because only 1% of the genes were actually looked at. So there's a lot more comparisons that can be made. So until the genome is fully annotated, no firm conclusions can be made regarding the improvement in annotation. But it's a promising step forward. So some future research examining which methionine is actually the true start of the protein sequence. So you can see here there's 3 different methionines in this new sequence, of which the third one is where the old sequence matches. So those new methionines might not actually be the true start codon, so that can impact our sequencing. We need to finish annotating the new assembly. We've only done 2,000 out of most likely 7,000 genes, so that's a lot more work to be done. Gather proteomics data. Proteomics data will help us truly learn about which proteins are expressed better and help corroborate our evidence. PacBio isoseq data was generated will improve future annotation work, and using the new annotation to improve research around the country. So finally, to acknowledge some people at the University of Georgia for allowing me to be here,
Thank
our
you.
youth program, especially Dr. Arnold, and all the members of our lab listed above.
That was wonderful work. Amazing what you did over the summer. I'm probably going to have to move on to the next speaker. Just you might we have that we have so we stay on schedule. And thank you so much. I hope anyone who comes up with questions will reach out to Miguel. This was a wonderful project. You did an incredible amount of work. So our next speaker is Jalen Reed. He's from Morehouse College. He also worked on the COVID-19 project. His mentor was Dr. Liang Liu, who was actually also a graduate coordinator in the statistics department. And it's my pleasure to introduce his work on SARS-CoV-2. Thank you, Jalen.
Thank you, Dr. Arnold. How's everyone doing? I hope everyone's doing good. So I had the pleasure of working with Dr. Liu, and we had my topic was phylogenetic trees of SARS-CoV-2 genes. So my hypothesis was, what is the origin of the coronavirus? I'm sorry. Just a little nervous. So for the introduction, the main focus of the research was to determine the COVID-19 origin by zoonotic transfer. There were 11 genes in the SARS-CoV-2 genome studied, as well as the sequence alignments. Gene trees were produced based on the DNA sequences to help show the phylogenetic relationships of the sample sequences using the 11 genes. So what are phylogenetic trees? So a phylogenetic tree is a diagram used to reflect evolutionary relationships among organisms or groups of organisms. And down here below are like examples of phylogenetic trees and how they are displayed. So about the virus. So SARS-CoV-2 is the virus strain that causes coronavirus. The SARS-CoV-2 has 4 structural proteins known as the S spike, E envelope, M membrane, and the N nucleocapsid proteins. And so if you can look at the illustration on the right side, the spike is what attacks the host and what really just puts the attacks the host really and puts the coronavirus in the person or whatever organism or whatever living thing it is. So the spike protein is responsible for allowing the virus to attach and fuse with the membrane of a host cell by binding to the receptor angiotensin, converting the enzyme to ACE2. So the SARS-CoV-2 genome. So if you can see in the red down here, these were the 11 genes. And there were a total of 30,000 base plates. And with that came from the Wuhan genome. And like I said, and there was a total of 29,903 nucleotides. So materials and methods. In order to do the research properly, RAXML and Figtree were 2 of the main computer programs used to construct the gene trees and visualize them. RAXML assisted the research by producing phylogenetic analysis from our datasets using its maximum likelihood algorithm, and Figtree helped by designing graphical phylogenetic trees based on the data produced from RAXML. So this is an example of one of the trees that I created or that I used. In this specific gene tree, bats can be seen as the origin for human coronavirus as its clade connects to the human sequence. So when I say clade, what that means is, and I made sure that you guys could see, if you guys could see it, the red right here is all human sequences. And then the blue down here are other sequences such as bats, such as bats and other sequences as well. And as you can see in the blue, if you look in this clade right here, the second biggest one that's connected to the red, as you can see, it is attached to the human sequences. So what that means is, that it will give, in a way, the bats were the origin for the humans to have the coronavirus. So in a way, like, I'm gonna repeat myself, I'm sorry. In a way, the bats in the blue down here in the clade, as they share the same clade, they each connect. When they connect to the clade that has the humans, this R down here, the red, is the human clade and human sequences. And when the bats are connected to that, that's basically saying that the origin of coronavirus came from the bats. Another example is my gene, or the gene ORF10. So in this specific gene tree, we see that the pangolin is the origin to the human coronavirus, as its clade is connected to the human sequence. So like I said before, if you look at the clade in the red, here are all the human sequences. But if you can look closely in the blue, pangolin is connected to the clade that goes to majority, well, really all the human sequences. So what that would tell you is that pangolin would be the origin for the coronavirus for the humans. And another one is the gene CoORF7b. And in this gene tree, bats again are to be seen as the origin for humans, as is located closest to the human sequence and share the same clade. So here are a summary of all the 11 genes, and here are the genes are all, well, here are all the 11 genes that I used in the origin as well. And this basically is to show the different genes and how they can give you conflicting results. So as you see, each gene has a different origin. And so and that can be another possibility for having different origins. Another possibility for having different origins is due to the missequences. And what I mean by missequences, so Raxamel and fig tree each are like an estimation. Now, due to estimation, there can be estimation errors. And through estimation errors, sequences can go missing and things like that. So in a way, it can give you conflicting results. So it's really not a stamped answer to is it the origin, is it the real origin for the human coronavirus? But I'm gonna give you more information to how you can get a more precise answer to that. So I'm gonna give you results. So like I said, there wasn't a direct answer to the specific origin of COVID-19. And the reason for this is due to the gene trees each being different from one another. In a way, it's like the gene trees each have their own story and tells a different story. There were also limited sample sizes for bats and pangolin sequences to work with in with the gene trees. Because the gene trees were in the estimation, the relationship between the sequences could be different. There could be estimation errors, like I said, or the genes could be truly different. However, one common pattern noticed among the gene trees was the coronavirus sequence RATG13. And so this virus is a SARS-related coronavirus found in bats. And based on the gene trees, it was linked to the origin of SARS-CoV-2. I'm gonna get into that in a minute. So with that being said, you really can't, like I said, it's really not a stamped answer to, okay, did humans receive the coronavirus through bats, pangolin? Because as you saw, they each gene showed a different outcome or a different way of humans being connected to a clade in a way. So, and over here is approaches to creating a species tree. And I'm gonna get into a species tree and what that means in a minute in the conclusion. But majority rules consensus method. So this is these are ways to make a species tree basically. And what a species tree is in a way a more in-depth of what your genes and a more in-depth way to see, okay, this is how coronavirus could have attacked to or been the origin to the humans. So majority rules consensus method. With my data, I was able to apply the consensus method to my gene trees. You can obtain a conclusion about the origin and species tree level, and the input data for this method would be the gene trees. Output data would be the relationship in the species tree. Excuse me. If the majority of the genes support a particular relationship, then the relationship in the species tree should be strong due to it being the majority. So with the 11 gene trees, the origin was examined, and some of the origins would be RATG13 or another origin. However, out of the 11 gene trees, RATG13 was the majority rule as it demonstrated a majority vote. So if we go back, As you can see, RATG13 is presented more than, you know, other origins when it comes to the genes. It's presented more as the origin for human coronavirus and how that can be connected to humans when it comes to COVID-19. I'm sorry. The coalescent model. So this was a model that Dr. Liu practiced on with his team. And so with this model, they used this to make a species tree. This is another way of making a species tree. And with that, they use RATG13 as the origin of COVID-19. Excuse me. And so another species tree method is the concatenation tree. And that's another way of making a species tree as well. So another approach is the concatenation tree. And the concatenation tree is just a tree for the entire genome. So this method is used when you concatenate or combine really all the 11 genes so that it will be the entire genome. And so this tree should be very similar to ORF1 due to it ORF1 taking up more than half of the genome. And I'm gonna show you what I mean by that. So right here. So as you see in the red down here, ORF1A and ORF1B each are, well, really, this is the entire genome that you're looking at. This is the entire genome that you're looking at. If you look at ORF1 and ORF1B, these two are the same. They just split them up, really. That's all it means. So when you look at it, what that's telling you is that if you use this whole genome, and you were to use if you was to make a species tree using the concatenation method, you would most likely get more origins from ORF1 and ORF1B. And that's due to because if you can look, you see how it takes up majority of the base pairs. If I'm saying that right. Base pairs. And as you can see, it takes up majority of the graph. So if you were to use the concatenation tree method, then you would most likely get ORF1. You would most likely get that gene tree due to it taking more than half of the genome, if you know what I mean. So conclusion. So once all the 11 gene trees were made, I analyzed each of them and noticed that RATG13 was not the origin for human coronavirus. And in order to figure out the origin, you need to look at a species tree and not an individual gene tree. I suggest that you combine all 11 gene sequences data and build a species tree. You cannot make an inference on the origin by just using a single gene sequence data. You have to use the entire genome to estimate the origin of the coronavirus. And just a little touch up on, because like I said, I had other origins for the human coronavirus, which were bats and pangolin. So the relationship between the pangolin and the bats changed across the 11 gene trees. Among the gene trees, there were different narratives being told. For example, in one gene, the pangolin would be closer to RATG13, while in another gene, bats would be the closest to RATG13. And so the reason for this would be either because the trees were being an estimate and there could possibly be estimation errors, or the genes are truly different. It isn't clear whether or not if there are estimation errors or if the genes are true or real due to the limited sample size. So if you were to take another sample size, the tree might change, the whole tree might change. And for acknowledgements, I just want to thank Dr. Arnold for this opportunity, as well as Dr. Liu for his help and support throughout the project. I was able to learn so much about the coronavirus and how it's really being done. And especially since, everyone was affected in a way, I was really in a way excited to see the behind scenes of how it all works to find out where the origin comes from. Dr. Lee really did help me a lot, and he did teach me a lot of things. And for one example, I came into the program not really having an idea of what a RaxML or Fig Tree was, but with his help and Dr. Arnold as well, I was able to learn a lot and navigate through this easily. But I really did enjoy myself this summer and just learning a lot.
Thank you so much. It was a wonderful presentation, and
Yes,
it's
sir.
it offers insights on to what the whole team was working on, and looking at what the individual, how the origin question varied across the different gene trees. Thank you so much. So, so you didn't have any prior bioinformatics experience, right?
No, sir, I did not.
Okay, so that's great. So good, and I particularly liked the analogy with each of the genes is telling us a different story of what happens.
Yes,
That's
sir.
a very strong analogy.
You could thank
So-
Dr. Liu for that, because that's
Okay.
like I said, he broke it down for me to where I could understand it, and that was
Right.
really how I grasped it.
Right.
I like to say this: when I was doing the presentation, while I was like conducting it, it wasn't until then I started really like knowing and really understanding. Okay, so this makes sense. Okay, this is how he
Right.
got that. And
Right.
Dr. Liu really helped a lot.
Oh, that's wonderful. He's a wonderful, wonderful teacher too, as well. I've seen him at work.
Yes, sir.
So I think what we'll do, we have two more speakers, and I want to try to sort of finish on time. If anyone wants to reach out to Jalen, you can probably send me an email and I'll forward it, or he's be glad to talk further with you about the project. And thank you so much.
Yes, sir. Thank you, Dr. Arnold.
Okay. And our next speaker is Lauren Stupp. She is a rising senior at the University of Georgia. Like Cameron, she's working on this fundamental symbiosis between fungi and plants, but she's gone in a different direction. Won't say completely different because you can't get away from bioinformatics. But she's really focusing on the bioinformatics aspects of this symbiosis. And on that note, I'll turn it over to Lauren.
Thank you, Dr. Arnold. I am honored to be able to talk with y'all today. My project is going to be focusing on utilizing neural network segmentation and classification to identify AMF colonization. So I want to give some background on some biofuel and using sorghum as a model plant. So biofuel is a great way to improve our nation's security and help mitigate the impacts of energy production and climate change. And sorghum bicolor holds a great promise as a model organism because of its high productivity, its high stress tolerance, and it's got a relatively small genome size, so that makes it easy for genetic analysis. It's also grows in drought-prone environments as well as low fertility soils, which means that it can be grown in numerous different environments. So with that, we get into the colonization of arbuscular mycorrhizal fungi. So there is a obligate symbiosis relationship between these two, in which the AMF cannot survive without its host roots. And in order to do this, there needs to be nutrient exchange. So the AMF gives phosphorus and nitrogen to the roots, and in return, the AMF receives sugars and lipids from the plant's photosynthesis. So This also increases the water intake from the plant and increases its stress tolerance. So it makes it great for those drought environments or very dry environments. So when we're talking about the obligate symbiosis, it's really important to notice that it's not just a fungus and root relationship. There's been studies where propagation has happened for super mutualistic AMF where there's just not consistent results. So this means that there needs to be a host AMF and environment communication. And this interaction is not as simple as just fungus and plants, which means that it's really hard to reproduce taxa that are going to be creating really high biomasses without introducing like a strict environment. So kind of want to change a little bit of topic. We're going to go into some deep learning. So what is deep learning? Well, machine learning uses algorithms to parse data, learn from the data, and then make informed decisions about what it's learned. Deep learning is then a subset of that in which it mimics the workings of the human brain in processing data to use for detecting objects. So what does that include? So it includes object detection, where the goal is to classify individual objects and localize using a bounding box. And then there's also semantic segmentation, in which the goal is to classify each pixel into a fixed set of categories without differentiating object instances. So what does that kind of include? How do you begin that? You need to train a pipeline. So a set of manually annotated images needs to get pre-processed to generate test and validation sets. And then a certain number of training cycles, also known as epochs, follow, where the neural network internal parameters are adjusted based off the test set. And then the validation set allows for the independent monitoring of the neural network preferences to be assessed by the misfitting of the model. And then those parameters that were best suited for the test and validation set are saved for future image sets. So what does a region-based convolutional neural network look like? So these neural networks are based off object detection models that use high-capacity neural networks from a bottom-up region proposals in order to localize and segment objects. It uses selective search to identify a certain number of bounding box regions of interest, and then it extracts those features from each region to independently classify them. So putting those two together, we wanted to determine how can we use these deep learning models to identify AMF colonization. So we talked about earlier how there's many different types of AMF because of the host, root, and environment relationship. So will the Detectron 2 or a deep learning system be able to accurately and precisely identify colonization when masking and classifying? And then also, how many images are required to train this system, because imaging is a lot of manpower, and will it be worth it to reduce the manpower on one side but put it more heavily on the other? So I just wanted to introduce the summary of our methods that got us to the point of utilizing a deep learning system. So in order to do this, we needed to grow the sorghum plants, and those were grown in the Iron Horse Farm in Athens, Georgia. They were then harvested and cleaned as carefully as possible so that the AMF colonization was not disturbed. Then the sampling, clearing, and staining process occurred, in which there was an equal amount of root put into each sample, about 2 grams. And then those cassettes were stained for AMF colonization so that they would be able to be detected in the imaging step. The imaging step included using a Primostar microscope with an XM camera at 10 magnification. And then we took those images that we took in the laboratory and uploaded them to the VVG image annotator created at the University of Oxford, and used a polyline to outline the segments of AMF and root that we wanted to annotate later. This helped with determining different structures of the AMF in the picture, as well as differentiating random lint or root from the AMF. And then finally, we are now utilizing a deep learning system called Detectron-2. And to begin using the system, we took 756 photos of AMF colonization to train the network model, which will then hopefully be able to predict AMF colonization and structures. So I kind of want to go over what Detectron-2 looks like, because it is not a classic neural network. So first, usually a neural network uses filters to extract certain visual features, such as like edges or arbitrary shapes from a group of neighboring pixels. And then the resulting feature map then feeds it into a pooling layer. The pooling layers are downsampled, aiming towards reducing the features width and height for the next convolutional layer. And then each layer tries to detect high order features through the rounds of convolutional pooling layers to identify certain features. These methods, however, in these methods, segmentation precedes recognition, which creates a slow and inaccurate process. Instead, this program uses a parallel prediction of masks and class labels, which is more simple and flexible. So in our method, we have the math, it's called a mask CRNN. And by adding a branch for predicting segmentation mask on each region of interest, so let me show you my mouse. So we feed in the feature map and we have the regions of interest here. And then from here, we have a break. We have the mask classifier, and then we also have your bounding box and object class. So this parallel system allows for a division. It allows for a deep decoupling of the mask and class production, which is different from other systems because usually it relies on the bounding box relies on the mask. So there's no competition in between the classes is what's important. And so moving on, we're going to look at after you run the Detectron-2, you get data that spits out in the form of an AP value and a mask. So the MAP value is what's used for object detection. It's the mean average precision averaged over intersections over unions, their thresholds. So you have a certain number, and it's basically determining when an object overlaps with another, what is the mean average precision of that? And that only computes for the recall values between 1 and 0, where the values can go from -1 to 1. So it's not counting those negative numbers. It's not wanting to count those. So when you're looking at AP values and when you're analyzing the data for this Detectron-2, you want to look at the different hyperparameters. So that includes AP50, AP75, AP100. And the AP50, you're going to look at for very large data sets, such as ours that had over 700 photos in it. And these are the intermediate results. You don't want to go over that because then it's going to overtrain the system. You usually want a range between 50 and 60. And as you can see in our results, we got a range between 46 to 39, which is still an amazing range. There was a set tested earlier that was between 25 and 20. So this has really improved our data testing. So I just wanted to give some examples of what this kind of looks like for masks. So we see the ground truth, and this is the manually annotated photo done by one of the researchers. So we have a root here and here, and then we have an internal hyphae right here. And when it's run through the Detectron-2 after it's been trained, you still get the root, but you get a little bit of different shape from the internal hyphae. And that's about a 74% accuracy, precision, which is still amazing. And it's very, very high tech, very state of the art. And the second example, we have the same thing, a root, and then internal hyphae that was annotated by one of our researchers. But here you see there's more annotation for other roots in the photo. In addition, this this annotation for the AMF internal hyphae is even slightly more narrow and more precise than our annotated one that was done manually. So how do we use the Tektron-2 to advance AMF studies? Well, we're going to use that to study the relationship between sorghum bicolor and AMF within a controlled environment. The crop's biomass and AMF colonization will be examined to understand the genetic variance among different genotypes. And then by utilizing the Tektron-2 or any other automated annotators, we'll be able to determine the colonization without having to do X amount of manual labor. This network would save time and money for laboratories, which would allow investment in further more projects or quicker advancements, scientific advancements. In addition, once more scientific advancements are made, there will be further applications for controlled environments within agriculture, genetics, and biochemical studies. And hopefully we'll be able to understand AMF a little bit better. So I just also wanted to note that there are other AMF annotators. There is Lauren, which stands for Laboratory Automation for Universal Recognition of Endomycorrhizal Features, and that was developed by UAU and Michael Scarra at the University of Georgia. And there's also the AMF Finder and AMF Browser developed at Cambridge University. And for my further studies, I would really like to see the differences between these three annotators and see which one is the most state-of-the-art for laboratories to use and get the most out of their machine learning. All right, and I just want to acknowledge my mentors. So Jonathan Arnold, Shufan Yang, and Yue Wu have been there for me all summer. They've given me so many opportunities and been so helpful in answering all my questions. Also, the Department of Energy, National Science Foundation, and University of Georgia have given me this opportunity through funding, and I'm so grateful for that. I just cited one source, and I would like to answer any questions you have.
Thank you so much for that wonderful talk. While we're waiting for some questions, I would just, in the initial greenhouse study, how many images did you guys end up putting together? You annotated 700 plus.
Yes.
But how many, if you were to do it manually, how many more would you have?
Well, in total, I believe there was over 21,000 photos taken as of right now. Last time I was in the lab imaging, I believe it was 21,000. And then I believe our goal was 24,000.
Right.
But to annotate all of this by hand, it took us about 2 weeks to annotate the 700 plus. I can't imagine how long it would take us to annotate 21,000.
And then don't you also have another collection that you're generating right now?
Yes. I am looking at the differentiation between colonization in the roots based off the location. So the middle, top, or bottom, and we've divided it to see if there's any correlation there.
But you're also doing something in the field too.
Oh,
right.
Yes. We're doing a GBOS study.
Right.
Yes. And that has been, I would say, manually the most challenging part of the summer.
Right. Okay. So I think we're almost on time. And thank you for this wonderful talk. And this is a coming together of two things, the bioinformatics and the systems biology of plants, that you can't do one without the other. And thank you for illustrating that. And so our last speaker is Tony Zhang. He's from Langley High School. He is following in the footsteps of Dean Dorsey, starting his research career in high school. And he's going to actually look at a physics problem. I'm hoping Dean Dorsey has come back in to see this. So he's going to look at the collective behavior of oscillators. And I'm turning it over to you, Tony.
Thank you, Dr. Arnold. Hello, everyone. My name is Tony Zhang. I am a rising junior at Langley High School, and I would like to present my findings on the MIND project. So first, let me give you some introduction into the project. The project was centered around the biological clock, which is a system that governs an organism's daily routine. So MIND stands for Maximally Informative Next Experiment, and the goal of the project was to discover the most practical nonlinear representation of the biological clock's phase synchronization. So what is a Kuramoto model? The Kuramoto model can represent the biological clock. It shows the operation of coupled oscillators which move in sinusoidal motion. The K, so here's the equation for the Kuramoto model. The K is the coupling strength, which influences how close or scattered the oscillators will be within the model. So with weak coupling, such as K equals 1, the oscillators were scattered around the model. However, with strong coupling, such as K equals 12, the oscillators were more in close proximity to each other. And N represents the number of oscillators in the model. I shows the individual oscillator numbers, and omega represents the angular frequency. In addition to the angular frequency, temporal frequency equals 2 pi times the angular frequency, and period is the inverse of the temporal frequency. In addition to the standard Kuramoto model, we can also add noise to the model, which accounts for randomness and desynchronizes the oscillators. Noise is added to the equation through a new variable called error, and this is the error variable. Now I would like to show you how the Kuramoto model was simulated in MATLAB. So 10 Kuramoto oscillators were simulated in MATLAB with and without noise over 320 time points. The K was set to 20, the N was set to 10 because of the 10 oscillators. The noise was randomly generated, and the oscillators were numbered from 1 through 10 so that the omega was set to individual oscillator number divided by 10. And here are the graphs produced by the simulation with and without noise. I'm going to put it up a little bit. All right. So next is the experimentation. So the simulation with noise was used for the experimentation and was run at different K values. The K value, for example, the first time this simulation was run, the K value was set to 20 as the original true K, and then the user would input their prediction of K, which in this experiment was 35, and also a K value was randomly generated. And the values from each of these tests were stored in 3 arrays, and then which allow, so yeah, and then the results were used in the subsequent chi-squared criteria. So what is a chi-squared criteria? A chi-squared criterion is used to find the discrepancy between the observed value and the expected value, the observed and expected value. And then 2 chi-squared criterion were used in this experiment, and the sigma was calculated from the observed value. So the first chi-squared criterion was used to find a discrepancy between the original K value simulation's array values and the values from the simulation involving the user's input of the K value. Next up is the ensemble method. So what is an ensemble method? An ensemble method combines several basic models to get a better prediction of the observations. In this experiment, the ensemble method consisted of a second chi-square criterion and a Metropolis Hastings Monte Carlo simulation. And then, yeah, the ensemble method was run 1,000 times according to the number of Monte Carlo steps, which was 1,000. And then so the first part of the ensemble method was the second chi-square criterion. So the Kuramoto simulation was run 1,000 times with a random K value generated by the twiddle. Twiddle is a variable which involves multiplying the specific Monte Carlo step width by a random number between 0 and 1, so essentially generates a random number. And the second chi-square criterion was used to find discrepancy between the original k-value and each k-value generated by the twiddle. And in the end, 1,000 results were generated by the second chi-square criterion, which I will also refer to as the new chi-square values. So what is a Monte Carlo simulation? A Monte Carlo simulation produces a probability distribution of possible outcomes. So in this experiment, the results of the two chi-squared criteria were used in a Metropolis Hastings Monte Carlo simulation. It was used to find the best ensemble of k-values with the lowest discrepancy, in other words, the lowest chi-square value. And each of the 1,000 values generated by the second chi-square criterion were compared to the single value generated by the first chi-square criterion. At this point, a new variable called x was introduced, and it was essentially a random number between 0 and 1. Another new variable introduced is called rho, which is generated by the exponential of the first chi-square criterion minus the second chi-square criterion value. And within the Monte Carlo simulation, the rho and the x values were also compared, and final arrays of chi-square values and k values were created, in which the value from either the first or the second chi-square criteria and either the user input k value or the twiddle generated k value were added depending on certain conditions, which I will get into the next slide. So here's an illustration of the Monte Carlo simulation. So essentially, if the new chi-square value is less than the first chi-square value, then the new chi-square value will be added to the final chi-square value array, and the twiddle generated k value will be added to the final k value array. The same applies if the new chi-square value is greater than the first chi-square value and the x value is less than the rho. However, if the new chi-square value is greater than the first chi-square value and the x value is less than the rho, then the initial chi-square value instead of the new chi-square value will be added to the final chi-square value array, and the user input k value instead of the twiddle generated k value will be added to the final k value array. So what are Monte Carlo step widths? A Monte Carlo step width is essentially the size of a Monte Carlo step. And in order to find the most exemplary Kiramoto model to represent synchronization of the biological clock, multiple step widths were tested in the ensemble method, which in this experiment were 1, 5, and 9. All these steps mentioned in the ensemble method, which I mentioned, such as like the second chi-square criterion in the Monte Carlo simulation, were done for each step width. Here are the results of the ensemble method. With a step width of 1, you can see that the chi-square value was the highest. It's because the final generated k value of 34 is the furthest from the original true k value of 20. However, with a step width of 5 or 9, the chi-square value was much lower because the final generated k value of 21 was much closer to the original k value of 20. And here are the graphs produced by the ensemble method specifically for step width of 5. As the method is generating k values, you can see that the chi-square is decreasing over time, indicating that the k values are getting closer and closer to the original k value of 20. As a result, a fixed step width of 5 or above recovers the true coupling strength and is therefore the best representation of the biological clock. Before I sign off, I would like to thank my amazing tutor, my amazing mentor, Dr. Arnold, for all the help he has given me on this project. He has met with me every week and has provided so much instruction to me, and I am truly grateful for that. And I would also like to thank Dr. Schutler for providing feedback during the clock meetings.
Thank you so much. That was a wonderful project, and it really gets at looking at the effects of noise on the clock, which is wonderful. And so the so you had to actually, what did you do you did all the programming for the Monte Carlo and the simulator. What did you do that in again?
MATLAB.
MATLAB. So, and did you find it was, you also had prior experience with R, I gather.
Yeah, well, that was last year, and I do a lot of Java coding, so I have a lot of experience with computer science, but this is my first time using MATLAB.
Right, right. So it made the project go really quickly, and the simulator code. I'm just talking until someone asks a question. The simulator code was general, right? You could have done. You wrote the function in MATLAB so you could do 20 oscillators or 100 oscillators, right?
Yeah.
Right. So that's pretty nifty, and so that gives you. I've got two questions for you. How efficient? This is from Dr. Kai-Xiang Yi. How efficient is your program in estimating the parameters?
It is, I will say, not the most efficient. Like with 1,000 Monte Carlo steps with 3 different step widths I tested, it takes like at least 7 minutes to get like the final table in those graphs. But with 100 steps, which is what I did most of the time when just testing out the program, it was decently efficient.
Yeah. So if you were to, just following up on Kai-Xiang's question, if you were to make it more efficient, what would you program in?
That's a great question. So wait, what was the question? Okay.
Yeah, so suppose you had started at the beginning of the summer and I said, okay, we can use any programming language you like. What would you choose to, and you had what you know now, how would you just make the run faster by choosing a different language?
I would probably use R because I remember when I used R last summer, it was super fast.
Okay. Do you think it would be faster in Java?
Maybe, but the problem with Java is that it's that you cannot like view like the table, like what's like in the tables and all that and the arrays with unlike MATLAB or R.
Okay. Okay, so this is a wonderful project. It really was a privilege to work with you, and I look forward to seeing what you continue doing. So it was a privilege to work with you, and I want to thank all of the supporters of the RU participants. They're not just the mentors here, but they're the people in the lab of the mentors and the college and the National Science Foundation. And please have a wonderful rest of your summer.